Yesterday, we heard a lot about the approaches in computational toxicology, including many of the databases. And today, we're actually going to hopefully, put all these things together and try to talk about what we're going to do with those databases in terms of the timetables, where we're going, the translational potential, and decision-making potential, which is really one of the goals of this workshop. Our moderators this morning will be Bill Farland and Lauren Zeiss. And Bill is currently Senior Vice President for Research and Engagement at Colorado State University in Fort Collins. He's also a professor in the Department of Environmental and Radiological Health Sciences in the School of Veterinary Medicine and Biomedical Sciences. In his former life, several years ago, he was Deputy Assistant Administrator for the Science in the U.S. EPA Office of Research and Development. And Lauren is Chief of the Reproductive and Cancer Hazard Assessment Branch of the California Environmental Protection Agency. We have the Midwest represented from Fort Collins. We have the West represented, and here we are on the East Coast. So take it away, Bill.
Thanks, Tom. Good morning, everybody. Well, Lauren and I have an interesting job this morning, because I think we had a very good discussion yesterday about some of the emerging science around computational toxicology, and really looking at this intersection of toxicology, systems biology, biostatistics, pure statistics, as we heard yesterday, and computer science. I think that one of the things that is going to be most important this morning is to really sort of delve into this issue of how this emerging approach to evaluating toxicology is going to inform us as we begin to look at both how we develop the science, and secondly, how we use it as we begin to make decisions about chemicals, whether it's in a federal context or whether it's in a private sector context. How do those data complement some of the things that we already do or we already have? I think one of the things that we heard yesterday was that there was concern about whether the science was ready for prime time. There was a question about whether or not we were trading something that we already knew for something that we were uncertain about. I think we need to be very careful about recognizing that the reason that we have approached this issue of computational toxicology and a toxicology of the 21st century is that many people have been dissatisfied with the kinds of things that we're doing right now and its ability to answer questions that we have. And so if there's opportunities for us to do something different and something better, we'd like to try to really explore that and take that on. I think George said it well when he ended up the session yesterday, when he said we have to be very careful that we're not letting the perfect be the enemy of the good, that we still have a long way to go with computational toxicology, and we are just now thinking about how to approach this and how best to apply resources to be able to move the science forward so that it can have an impact. So with that, I want to start this, what we've called a structured roundtable discussion this morning with our practitioners from the Federal Liaison Group. I'll take a moment just to introduce individuals that we've asked to help us to address some of these questions. On your agenda, you see a whole list of questions here, and we have identified individuals to at least start the discussion here. So let me just introduce the discussants that you have on your list, and we'll go from there. Down to my right is Dan Axelrad from the US EPA. Morning, Dan. Gail DeBoard from the NIOSH program, Centers for Disease Prevention and Control. Good morning. Tina Levine, across from me, from EPA. Trey Thomas from Consumer Product Safety Commission, up at the far end of the table. Richard Judson from US EPA, and we heard from Richard yesterday as well. Bruce Fowler across from me there. Bruce from Agency for Toxic Substances and Disease Registry. Chris Portier is with us. Chris, this morning we'll ask him to put his NIEHS hat on again and make some comments as well. And Bill Setti from EPA. Bill is down at the end of the table down there. And Richard Beeger from FDA. Richard, they're across from us. So we've got a good group of people who are representing a number of different federal agencies. Most of them have had the opportunity to hear the discussion yesterday, and I'm sure have some thoughts. So, Lauren, I think we'll just jump right into the discussion. The first of the questions is, what are some of the strengths and weaknesses of the approaches that we heard yesterday? And I think Bruce Fowler is going to start us off with just some opening remarks, and then we'll open it up for a conversation around this topic.
Yes, thank you, Bill. I'm like George Daston, I'm an optimist, and so I was very glad to have a chance to put before you some things that I believe are very positive aspects to computational toxicology. And I have 5 minutes and 3 slides, so I'll warn you of that. Okay, thank you. Okay, so this is just a laundry list of the things that I've thought about and that we've had experience with from the perspective of people who use computational toxicology or some of those tools for risk assessment. First, I think most of us could agree it's fast, it's cost, it's timely, and it's timely. So you can have rapid screening of mixtures in, for example, emergency response situations. The one we used this for was actually right after Hurricane Katrina. And the question was, how do you prioritize cleanup of some 10,000 chemicals in a place that's completely devastated? So our folks did essentially QSAR studies and tried to give FEMA and the other people a laundry list, a hit list of how they should prioritize what they were going to do. The ability to handle large and complex data sets. Now, we talked about this yesterday. We didn't get into the molecular docking studies, which I happen to be particularly jazzed about as a set of techniques, but it's a way of basically screening through chemicals and looking for those that are most likely to interact with the biological systems and then put those forward for further testing. So a way of narrowing the playing field, if you will. Expeditious sharing of data sets. CompTox lends itself to shipping large data sets around and allowing multiple users access to those data sets. So we've been working with FDA and NCTR on a couple of projects. We have a CompTox colloquium group, I'll call it that, or consortium at this point, between ourselves, EPA, NIH, FDA, and we even have some people from the Army in there. We keep picking up people as we go. The next one of these will be sometime, we hope, in December or maybe January here in Rockville. Chris Austin agreed to be the host. So if anyone's interested in that, it's open. We're all volunteers welcome. Risk assessments for population at risk. Now we talk about this, age, gender, genetic inheritance, but how do we get at that? Okay, how do we translate the basic biological or molecular data into actually using it for risk assessment? And finally, the capacity to interface with chemical and biological endpoint data sets, so exposure analysis, in vitro data, in vivo data. I've got a couple of comments about that in the last slide. Let's see here. No. How do I advance this? Right. Oops. There we go. Okay. This is something I think we all recognize intuitively, that within a population of individuals who are exposed to a particular chemical or have a particular blood concentration or urine concentration of some substance or group of substances at issue, there's a subgroup, a sensitive subpopulation. And we talk about them in terms of things like age, gender, nutritional status, genetic susceptibility, and mixture exposures. We touched on those yesterday. This one here is one that I think is particularly of interest because CompTox allows us to begin to incorporate or superimpose things like SNPs analyses on tops of exposures, and in terms of helping to better define or more precisely define those people who might be at special risk because of how carefully they chose their parents. Now, this is the last slide. basically the idea is that you can move from in vitro or in vivo studies up through various levels of biological organization to actually help conduct human risk assessments to improve the precision of those judgments. Now, the thing I would point out, and Debbie Clory-Schlechter brought this up yesterday, is that in order for all this to hang together, we have to, the collective we, or as we say in Georgia, all y'all, have the capacity to trust that this computational number, whatever it is we come up with, is in fact linked to a biological reality. If you look at this line of things, you actually can run the equation the other way. You can run the paradigm in reverse. So you can say, well, okay, the computational toxicology gives me this particular answer, this judgment. Is that really true? And can I go back in relatively short time at relatively little cost and verify yes or no whether or not this holds water? I think you can. That's the optimism. And I think there are people in the pharmaceutical industry who are doing things like this right now. So those are the things that I see and that we believe are positives about incorporating the computational toxicology into risk assessment. Stop there.
Some comments or thoughts about these opening remarks, or just some general issues about some of the strengths of the science that you heard yesterday?
Sure, I'm particularly actually encouraged by this comment about combining like SNP analysis, like polymorphisms, populations, genetics, with the in vitro data. I mean, with this kind of computational data, I think if we can do that, then I think it really added another layer of confidence into this kind of research.
I agree. There's a paper published by Manhui Chang is her name. It's in the American Journal of Epidemiology about January of this year. And Manhui is at CDC in Atlanta. She mined the NHANES database for probably about 30 enzymes, 30 genes in their SNPs. And these are human data that are available from the NHANES. There are more in that database that just have to be pulled out yet. But the idea is that you actually could have the human SNPs available, and you, as you just said, you could then take some of this and put it back into some of these in vitro systems and actually test the impact of a particular SNP on the response, whatever the biological organism is.
Bill mentioned when he first started about how we ought to be thinking about this as a tool to advanced areas that have been perennial problems in toxicology. And Bruce, in his first slide, and the first line of his first slide, mentioned mixtures. This is an area that we've always had trouble with. And it seems like we certainly can use it. He's certainly used it in this rapid screening context, but it seems to me that it also has some potential for helping us understand interactions between chemicals. And I think that this is an area that we should be going from the molecular level all the way up into the databases to try to develop a better conceptual framework for and do the testing necessary to determine the mechanisms of these kinds of interactions.
Thanks, Sarah. I think there were a number of occasions yesterday where it was pretty clear that we would have the ability to look at a defined mixture of chemicals or even a complex mixture that we might want to know something more about, and that it would fit very well with that type of a system, unlike the situation that we typically have when we do traditional toxicology testing. George, did you want to jump in at this point?
I had some, sort of a different aspect, but maybe it's the same sort of thing that we're talking about, which is, how can you use this in practical circumstances, which I think is what the Bruce's first bullet point here is. I think that this is a great use of information. I mean, one of the things that we sort of lost somewhere along the way in applied toxicology is the idea of hypothesis statement and testing. And we have the ability, really, with the historical database that we have, which is what I think Bruce is relying on there, to actually use the wealth of information in a computational way to make inferences about the toxicity of untested materials. So, one of the things that Richard brought up yesterday was the DSS tox data set, which has tens of thousands of entries for a whole variety of toxicity endpoints. And it represents what's out there in terms of the published in gray literature on the toxicology of chemicals. So you have the ability with that DSS tox program to search chemical substructures. And so if you have a new chemical, but it has a chemical substructure that you, with expert judgment, would infer would be biologically active, or you can search something that would be a likely metabolite of your new chemical. Those sorts of things would allow you to understand what's out there in terms of analogs in the literature that might inform you about the toxicity of a new chemical. Doesn't mean that the new chemical or the untested chemical does have those properties, but it allows you to start to state some hypotheses. And those hypotheses might be as simple as, if new chemical X is hydrolyzed by esterases, it will form the two chemicals that we know an awful lot of the toxicology about. And this allows you to move forward a lot faster. In the case of an emergency response, you're just going to have to live with the inference. In the case of where you can consider things more, your toxicology becomes a simple exercise in the lab in an afternoon, rather than something huge. So these are the kinds of things that are actually available to us today. I mean, you need the database, which already exists. You need the computational skills. You need some sort of expertise in chemistry. And either you have a chemist on hand who helps you, or some of the things that Alex brought up yesterday in terms of OECD Toolbox actually have those capabilities to help those of us who are less capable in chemistry work through those things. I think these are the kinds of applications that we can do now. I think that they're great examples of what computational toxicology can do. These are the things that are going to stand us in good stead as we start to develop the much more sophisticated projects that we heard about yesterday.
Thanks, George. I think that that approach that you just described is really one of the strengths of this sort of shift in paradigm that we've talked about, and I want to explore that a little bit more. One of the topics that we have on here is identification of stressors and how we would use this to do that. I'll just put a placeholder out for people to think about. Chris raised the question yesterday about should we be focusing more on human information as we draw those kind of inferences, or should we be going back and sort of matching against the toxicology database that we've had in the past and asking whether that's our gold standard for making those inferences. So we'll get into that a little bit more when we talk about identifying hazards, but let's at least keep that thought that George raised.
And I just wanted to add that the two are not mutually exclusive.
Mm-hmm.
Thank you, and I don't want to touch that topic that you just raised just yet, but instead build on some of George's remarks and taking as a springboard Bruce's points up here in terms of how can CompTox be used absolutely today on the autumnal equinox in 2009. And And I think the point that you raised about hypothesis testing and hypothesis generation really sings to me as a person in a regulatory office. I can certainly see the use of CompTox and all of the tools that, and excuse me for having missed off most of yesterday, but I did the cramming in the paper. The tools that were raised in a lot of the papers in two ways. First of all, in terms of the chemical structural alerts that you mentioned, and in particular, when I was cramming for the session and reading the paper by Zhu, I was struck by the list of structural alerts that came up, at least in this particular study. And I think this would be an amazingly useful tool in terms of emergency situations, prioritizing of chemicals, and so on. The other place where I would very much like to see more use of CompTox and all the high throughput tools is in generating hypotheses about sensitive human subpopulations. I certainly think we're at a point now where we need to be moving away from assumptions that a population may or may not be sensitive to a particular spectrum of chemicals, but rather to formulating those assumptions as hypotheses and going out and testing them.
Thanks, Rita. Chris?
Thanks. I also think it's kind of intriguing to think about the mixtures issue, that computational toxicology might give us the power to look at not only how different chemicals might interact with a single receptor, but along multiple points along a toxicity pathway. So you might have one chemical that interacts, upstream, another downstream along a pathway, and how that might contribute to a toxic response. And when you broaden it to think about the perturbing toxicity pathways, you might have very different chemicals interacting at different points along those toxicity pathways, or at points where different pathways intersect or interact. And I think it just could give us just more tools to explore this issue of the complexity of mixtures.
Okay, I'm going to rain on the parade. The question was about strengths and weaknesses.
Strengths.
Strengths?
We haven't talked about weaknesses.
I'm about to talk about weaknesses. I thought, oh, okay, then never mind. I thought we were doing.
Yeah, thanks. I guess to me, this might sort of be a bridge to the weaknesses, but I think some of the discussion yesterday, to me, left real major questions about how to even think about this question of strengths. I think there's still, to me, some unresolved questions about what the vision is and whether the vision is still the same as what the NAS committee laid out in 2007. And I heard some of our speakers yesterday make comments in the discussion directly along these lines about what are we really trying to do? Are we moving towards using high throughput screening data and CompTox for risk assessment directly to replace animal testing, or are we using it to really prioritize where we're going with whole animal testing? And I think you could see that it's both, but I think the relative emphasis between these really makes a big difference in how we think about strengths. And I think that's still an unresolved question, which sort of surprised me from yesterday's discussion, that some of the major players in this field expressed some uncertainty about where they're heading or what the relative emphasis is between whether it's replacement for whole animal testing or prioritizing whole animal testing.
So that clearly is one of the topics we wanted to try to discuss this morning, and I think we'll get into that as we move down our list of questions. Richard, let's hear your comments, and then we'll move on to Tina.
Yes, I want to talk about the sensitive population that Bruce brought up. I think that the SNPs are going to play a huge role, but also I think there's going to be a lot of age and gender and that kind of information can be picked up by metabolomics. And I just think that is, if you believe that genetics is going to solve that whole sensitive population, it's only going to be probably true 10% of the time. It's the mixture of genetics with your nutritional status and your age that's actually going to provide what your sensitive population is. And I believe metabolomics is going to show you the way on that.
So I think that's probably a good reminder that although we had a good overview of various aspects of science that feed into computational toxicology yesterday, because of the time that we had available, we didn't hit all the topics that we might have, and clearly there are other areas, including metabolomics, that will feed in. And as we get further along with the science, I suspect there will be other areas that we're going to want to pursue. We've already looked at the evolution of approaches to doing various genomic types of screening, and I'm sure that those are going to continue to develop as we put more questions on the table for this particular topic. Well, let's make a transition into a discussion about limitations and weaknesses, and Tina's going to lead off with a few comments to start. Even
yes,
though I
don't
know
have
you're
any
an
slides, but I have a few comments. And first of all, I want to say that I think when Abby's first statement yesterday afternoon really did sort of echo, I think, my feelings and certainly OPP's feelings about tox testing in the 21st century, about the NAS report. I think it's the vision is an extremely exciting vision. And I think that rather than talking about some of the things that were talked about yesterday, you know, issues of metabolism and dose response and those kinds of limitations, I'd like to talk a little bit more about maybe ways in which we could be limiting it from reaching its full potential for the area of toxicology and for the area of safety assessment. And I sort of want to wax nostalgic a little bit and remember when I started graduate school in toxicology, which was, I hesitate to say, 40 years ago. And the first toxicology seminar we had was on air pollution. And I was actually an experimental psych major that was in a fairly new field for me, and went into this room and there were a bunch of topics on air pollutants, and people picked topics. And then they would basically describe what the given air pollutant did as part of the, you know, and I was sort of into like, well, I want to know how it does it and what the mechanism is. And there really wasn't a lot of emphasis on that in those days. And then I went off and had a regulatory career. And for a while I was a regulator, and I didn't go to SOT that often. And maybe 20 years later, I came back to SOT, and I said, oh my God, I'm so out of it. You know, there's apoptosis, and there's, you know, oxidative stress, and there's all this molecular biology, and all of the graduate students that graduate that we now hire are not whole animal people. They're people that have worked in these kinds of fields, and it's a totally new field, and now everybody's into the mechanism. And you have to remember that the safety testing stuff happened years ago, and there actually is, I think, a clash of cultures here. I think if you think about the way regulators think about toxicology, it's a let's find the NOEL and divide by the safety factor and come up with a reference dose. And I can even remember discussions about we don't need to ask for that test. We don't need to know what it's doing. We just need to know what the NOEL is. And this is not what we do as regulators. And I think that limits us so much as regulators. I think that if we really want to protect the American public, the vision of this is that we could begin to understand what's happening and have a much better way in which to protect the sensitive people and get a sense of where we need to be looking. And so I really think we need to think of this vision as a tool to bring the whole area forward, and not necessarily have it replace what we have now in a very hard and fast way. And I think we need to be careful about the way we start developing these things. Because, for example, you take the ToxCast project, which is being sort of based upon the best data we have, much of which is in the annals of the Office of Pesticide Programs. But you have to understand that in general, the testing that's done in the Office of Pesticide Programs doesn't really look for tox pathways. And so we may need to go elsewhere. I think that the idea that we're going to some of the discarded drugs where the drug companies are really targeting a pathway and looking for information on that pathway may be a much more, may yield more fruit than the pesticide stuff, because if you're trying to build some sort of tox pathway vision on data that's largely, body weight changes and apical measures, it might not really work. So I really think that maybe one of the biggest limitations, and if we can identify it and get past it, is let's not just think the same old way about testing, and let's start to use this whole process to bring us to a better place in science. And I think by extension that the regulatory science will be in a better place, because we'll be basing it on a much fuller understanding and a much better sense of is this relevant to humans? Which endpoint should we be most concerned about? And so that's really, I think, the message that is maybe a little different than other messages that I wanted to start the discussion with. Thank you.
I wanted to build on that a little bit and to say that I think that the predictions that come out of CompTox can help to tailor basically testing, and I think that's sort of and this is a concept I've tried to move with for a long time. I mean, a lot of these pesticides, the safety assessment list is sort of a fixed list, and there's tension because it grows and so forth. But as Tina says, it's not tailored. There's a whole new generation of materials that pesticides encountering. Glufosinate ammonium is one of them. It's a glutamine synthetase inhibitor. Well, we don't have any testing for that. And so simply, that's an example of simply by the chemical structure and by what in fact is research that people have done, you could make you might want to then be concerned about things that look like glufosinate ammonium, and there are others, the whole rotenone story and complex 1 inhibitors. And so you can use both what George was referring to as sort of existing knowledge in toxicology at a more specific level to tailor the kinds of testing that you might want to engage in. So
one thing that my global talk didn't sort of get to is because there are lots of things that we know that the kind of testing we do now isn't particularly good at picking up. Parkinsonism, the animal models aren't very good at it. So I do think that we may get clues, and we won't get those clues if we only base what we're doing on, what we currently have. So I really think we have to mix it up a little to bring everything to where I think it ultimately can be.
Well, I actually have a clarifying question. So if I understand what you're saying, say you have an in vitro test battery and you get some positive results, and you're comparing it against standard animal testing that may not have looked at that endpoint. And so, which is right? The in vitro test that yield a positive result, or the standard animal tests that actually are negative, but because you haven't even looked at those endpoints? Is that the issue you're raising?
What I'm saying is we shouldn't necessarily assume that that's because something doesn't match up to what we have today, that there isn't value there. And I think the real value from this whole process will be for us to continue to pursue and follow up on and do the research that's necessary to get to the best answer.
Okay, now I can go through my list. I'll start with, yeah, actually very well. I'll start with the first one on my list, and I think probably the biggest weakness of what I saw, and that is there's no clear purpose outlined for what's being done. It seems to promise everything. Nobody's drawn bounds around it. We constantly go off on tangents and talk about susceptible populations, oh, we can do that. Dose response, oh, we can do that. We can do everything. And the truth of the matter is, we haven't done anything yet. I want to see a plan, a step-by-step plan that's going to take us somewhere in the use of this material in risk assessment and for making decisions about public health. I don't want it to promise everything. I want it to do one thing, then do another thing, and show me that it can work. And I'm not seeing that here. I'm seeing a promise of everything. When we at the NTP started this in 2003, we had a clear idea of what we wanted to do with this. We still have a clear idea of what we want to do with this. We want it to give us priorities. That's our first and foremost need. Once we have priorities, we'll take it into testing. Our second need, second clear use of this is mechanism. If we can judge mechanism, then when we go to do further testing, we can tailor the testing on the mechanism. Maybe doing tests that don't use as many animals or don't use animals at all. Third level is maybe we'll get something about dose response and some idea of potency from this. Fourth level is maybe we don't have to do any animal testing. But I think we have to take this one step at a time. And I'd like us to address a reasonable first step. So that's my first problem with what I've seen in terms of what we're doing. Second is, we seem to be looking under the light. Where's the science behind the targets we're trying to get at? We talked about hypothesis-driven issues, but where's the hypotheses? We're dealing on such a big, broad level that we're not necessarily focusing on why we're doing what we're doing at this big, broad level and what we're going to focus on doing as we go along. In the overall analyses that we've seen, I see a lot of anecdotal analysis. I don't see very strong, unbiased scientific analysis. Oh, we saw it hit this pathway or hit that pathway, it must be right. Gee, it does this. Gee, look at this chemical. It worked for this chemical. It must be right. That's much more anecdotal evidence than a very careful evaluation of the evidence in front of us. There are computational and analytical issues that are being glossed over in doing this. Abby pointed out yesterday, garbage in, garbage out. Some of the things we're looking at, we have no idea what the analytical problems were in the development of the thing we're using to go a step further. Just looking at the high throughput screening that NTP does, it took us over a year to figure out how to deal with a plate bias. There's clearly a problem with the plates on these analyses. It took us almost 2 years to figure out what to do with the worms and the worm tox in dealing with the well effects and some of the problems associated with looking at worms as they run through scanners. There are technical issues involved here that we haven't worked out that I see as a severe weakness in where we're going, and we're not necessarily discussing those technical issues openly and honestly with the community that's going to use this information to make public health decisions. There are sample size problems in what we're looking at. It's great that we want to do 10,000 compounds or 100,000 compounds. That's fantastic. But again, going back to Abby's garbage in, garbage out, there's a trade-off between how many chemicals I do versus how many replicates I do. And if the information I have is not sound and secure in the sense that the signal exceeds the noise on the individual inputs, I'm afraid that the thing on the tail end is not going to be very clear to us. And I think we have to put together the little pieces to make sure that in every assay we do, whether it's a high throughput screening assay like done at NCGC, or whether it's a toxicogenomics or a metabolomics assay, we have to look at sample size and seriously consider whether the thing we're getting, the signal, exceeds the noise. That's a really simple, important concept, and I don't see that being looked at. So those are the weaknesses I see.
George, and then Rita.
I think that those are great points to bring up. I do want to take issue with you about the we don't know where we're going kind of thing. Perhaps we who constructed this particular meeting didn't set out enough of an agenda for that, and I'll take responsibility for that. But it's not just the NIEHS that has a real strategy. Certainly, EPA's computational toxicology program has a real strategy that has clear goals in mind and has for the past 5 years. Certainly, my company has clear strategies. All of the pharmaceutical companies that have done endeavors in this have clear strategies. They might be different. And I think maybe what you're asking for, short of having some sort of czar, is some sort of a compilation of these things to see whether there are aspects that we're missing. So, I think that for thank you.
George speaks and music starts.
I know, it happens every time. I should have a soundtrack. It's like the pirate in Peter Pan. So I think that's what I would take issue with. In terms of the a lot of the technical aspects that you bring up are really the wet lab stuff, and I think that we could spend a long time talking about that. I think what we really wanted to stress more than anything else in this particular workshop are the computational aspects. I mean, what's different now than what we ever had before was the computational power to actually do very complex things and to take our reductionist biology of the last 50 years and start to make sense out of the messy wiring diagram that Steve Wiley had yesterday. And that's what I would hope we could focus on a lot more in this workshop than a lot of the admittedly important wet lab aspects that you're talking about, but maybe those are things for another day.
George, I'll just make a comment that we really didn't present the full EPA CompTox program yesterday. We touched on a number of aspects of it, and there are quite a few publications that talk about that, and people should be familiar with that. But I think a number of the agencies are actually making investments in these sorts of things, and I agree with you on that. Bob Cavlock would say that. Bob is actually up testifying at OMB today, I think, so he's not with us, but he would certainly have seconded that. I think the other thing is that another issue that Chris raised had to do with this question of whether a lot of this is ad hoc and anecdotal. And frankly, I think the program that Bob has put together and the people that are looking at some of these things have really taken an interesting approach, because we heard from Richard about some of the sort of bottom-up type of activities that have been going on within the CompTox Center, but there are also some very good publications and examples that have come out of there that have really been top-down, that have said, let's develop a hypothesis around a mechanism and let's really go back and explore some of these things to understand if we can really get a better sense. And so data that he's published on a number of the pesticide mechanisms of action fall into that kind of a category. So I think those are aspects that we need to think more about. We had Rita and then Yvonne, Mark, and then Abby.
Well, I certainly want to thank Chris for his list/rant, because I think there were some extraordinarily useful points in there, and I appreciate your remarks as well, George, because in truth, in my situation as someone who needs to ensure that the science supporting regulations is in fact supportable, I have heard many sell jobs, if you will, on CompTox as the absolute panacea to all my woes, and it'll eliminate all the need for animal testing in your lifetime, if not next year. And I do tend to view any sales on a panacea with a very jaundiced eye at this point. And the extent to which I hear plans and see a comprehensive approach, or even better, Chris, do see some successes. We have actually done this, and now we're going to move on to step B. I am much more happy with that. But what I actually raised my card for was to go back to some of what Tina was saying, which is again, in my position as a regulatory scientist, I need to ensure that the risk assessments that support the science, support the regulations are in fact supportable. And I always like to go back to the remark that no risk assessment has ever improved public health. No risk assessment has ever saved a life. You have to do something as a consequence of that risk assessment or in conjunction with a lot of other assessments that you will be doing. To that end, I think it is extraordinarily important that our risk assessments be as reflective of real life as we as scientists can possibly make those risk assessments be. And to me, at this stage in the evolution of risk assessment, that means understanding to the best of our ability what does the chemical or group of chemicals do. What is the mode of action? What is the mechanism of action if you can do that? Call it a toxicity pathway or a component of a mode of action. To me, it is getting to the same thing. What is this thing doing? And how can I counteract that doing in the environment? And this is the place where I think we can really capitalize on what is being learned right now from the high throughput assays, tie them back to granted our standard toxicity testing, and also use that to insist that any future standard toxicity testing or non-standard toxicity testing be focused to finding out what the chemical does. Even if we don't have the mechanism to understand what it does today, we may be able to do that in a year. Hence, slice, dice, keep every possible piece of tissue and sample, and use what we can in terms of these more molecular approaches to formulate some study designs and study types that will give us the information that we need in the next couple of years. End of sermon. Thank you.
Yeah, I would like to thank Chris for his, great outline of challenges. I wouldn't probably call those weaknesses, as we can clearly strive to address those rather than declare that it's impossible and then move on to something else. I do agree that no clear purpose outlined is the greatest challenge, but this is the challenge that the users, the end users of this technology, the government agencies, both EPA, ATSDR, FDA, and NTP and IEHS, will have to assist with. So Tox 21 is a great intergovernment agreement where there's a lot of discussion as to how to use some of this data and where to go and how to outline or at least align the purpose or the vision that each agency may have. The point of presentations yesterday was to, with a very kind of, not the 30,000 foot view, but at least the 10,000 foot view of where the field is. And so it is easy to point to some, clear weaknesses or challenges that can be addressed in the particular science that was outlined. But I was hoping that we can, take several steps back and understanding what can be done, we can then outline what should be done. And I think I would be in strong favor of NTP and NIEHS, again, and EPA outlining where the needs are, so then academia and industry and others can go out and get that information. Because right now there's, we're trying to do everything. You're absolutely right. We're trying to fill the elephant from all possible directions. But if you or others would like to form the sets of criteria or define what questions you want the new science to address, that will make life easier for people who are trying to do that research, but it also will help us to manage expectations, because I agree with Rita that it's, right now this is being touted as the new or next big thing, and it shouldn't be, because as every new science, it has its own limitations. But we do need to be very careful with this particular situation, not to turn it into, again, something that we were all experiencing, 5 or 7 years ago with genomics, where that was going to replace all the animal testings for $500 and no animals. I do play that clip of one of the former directors of NIEHS outlining that vision to my students in my class, saying that this is where the director of NIEHS projected in the early 2000s that toxicogenomics will take us, and we're still not there. So I think we need to be very careful with, how strong we want to pound on weaknesses, and whether we want to, just, work really hard to address those without, turning this into a slugfest and just, having the public and all the end users, disconcerted with where we are and how weak the science is. We shouldn't, throw the baby with the water.
I think Chris put out a challenge basically to parse that in a way that there are specific issues that we can address, and they'll be related to improvement of the science or application for priority setting, or eventually getting to some of these other issues. And the question is really, how do we all reach that kind of an agreement and move forward to take that first bite at the apple and really demonstrate success. I have Mark, Abby, Chris, Martin, and Richard on the list here. So, Mark.
Thanks. I'm very excited for what I saw and heard yesterday. I see a lot of interesting tools with a lot of very interesting applications. I feel like I have this broken car and someone gave me this credit card and showed me the Craftsman tool section in Sears for the very first time. I just don't know which tool to pick up first, And so I'm really concerned that we don't put the cart before the horse. I want to make sure that we use the right tool for the right job, and we don't do what's cheap and easy first, as we often do in a screening approach. I don't really see an alternative to animal testing, because we really still have ADME to deal with, exposure, species differences, genetic polymorphisms, and other things. It seems to make sense to maybe start with a QSAR, move towards in vitro. They'll help focus our in vivo work, and then maybe through to in silico, where we do more of a mechanistic approach and refine our mechanisms to understand how it's going to affect humans. But I would really like to see some kind of a paradigm set forth where we can see where these tools actually fit in, the questions we're trying to ask and answer.
Say amen to Chris. And I was really pleased to hear you identify a number of things and to talk about how at the current literature, I would say particularly more in the toxicology than in the basic research, there's an already assumption that if I see something in vitro or in the mRNA assay, it was really interesting because the question that was raised was, if I see something in the in vitro, which's true, the in vitro or the in vivo? And I think the problem is that I actually might err on the side of the in vivo until the in vitro can actually be validated with something that follows up biologically that can help validate what I saw in vitro. So I was wondering, in the first baby steps that you take, and George, I actually am concerned that we talk about computational toxicology as if it's something separate from the data that you have to enter into it. And I think that it's all really, that it's the data that you enter in that's what's going to make computational toxicology work. And if you don't get those basic data of what's a good screen, what is predictive, and do that very tedious validation work, which nobody in NIH wants to fund because it requires looking at both negative data and positive data, that's where I think funding needs to go, at that basic level of generation of those assays that eventually will be put into the system from which we build computational toxicology. Computational toxicology is only as good as the data and the assumptions that are made. It's not separate. And so I can't think about computational tox without thinking about the issues that Chris has raised. But I'd like to challenge you to think about risk assessment a little bit earlier in dose, because I think you are focused more on hazard identification and saying, okay, can I see something and test something, and then, but I don't think we're going to get to prioritization very fast if we can't also incorporate something about dose. And I'm hopeful that computational toxicology will allow us to make that happen. So I just wanted to ask you whether you could rethink that and put that early on rather than later.
I think we'll probably talk about dose and dose response as we get into the discussion today. But I do think that this issue of what is the standard that we're going to sort of measure these inferences against is going to be a good one for us to talk a little bit more about, because that is a challenge for us. Are we going to go back and say, you know, gee, we understand some of the biology, but it doesn't match what we saw in a whole animal bioassay, even though we know the limitations of that assay. So
Well,
I think we have to explore that a little bit more, and I want to do that.
Yeah, and everyone in the genome area, maybe I should ask these guys to speak up. They won't make a statement or a conclusion. They say what their research says is a hypothesis that then they go test functionally before they believe that data. And yet we're, in the toxicology world, really eager to make big assumptions. And I think that's where I'm really concerned, because then we're going to just have a system that's very weak, that it'll just collapse upon itself.
Well, I think there are clearly checks and balances in the regulatory arena that don't allow us to make those assumptions too quickly. So for those of us who sort of labored in the field of trying to get mechanisms accepted over a number of years, it took a long time. At any rate, we've got a whole list of folks. Chris, you were next. We are going to try to dial back in to get some of our folks on the line. So pardon the sort of interruption as we're doing this. Chris?
I'm glad I stimulated the conversation. That's very good. And I don't necessarily believe everything I said. I really wanted to stimulate some conversation. I do want to point out that my intention was not to throw this out. Clearly, I'm a strong believer in the acquisition of this information and the use of this information in helping to drive public health decisions. There's no doubt in my mind that the type of information we're going to get is useful. But I'll tell you a short story. For those of you who have followed the dioxin issue for years, TEFs has been a major issue with dioxins. So you've got this whole class of compounds. They have different potencies, and you try to figure out what the potencies are and whether or not the whole concept of TEFs will work. So the NTP decided to address this question, I don't know, 15 years ago, 12 years ago, a long time ago. And we sat down and designed a series of studies to address the question of TEFs. And while we designed the studies, we designed the analysis for the studies. And when we finished the studies, we analyzed it the way we had decided to analyze it in front. And when we were done, we had one of the most definitive statements on TEFs that could be made in the toxicological literature. And it's because we designed it to address the question at hand, and the analysis was designed with it. What I meant by we have no particular direction going here is that there does not appear to be an analysis tied to the strategy of data acquisition. What that tells me what the optimal design is. It tells me what's the optimal information to achieve. And we don't seem to have that worked into this very well. On the NTP side, for the data we're looking at and the data we have started to collect and the entire roadmap we put together, we had that. We knew what we wanted to do. And we still have that same concept of going through the idea of looking for pathways that are affected by the exposure to the compounds, targeting those pathways, and seeing if we can use that to drive the result. We may use different tools to target those pathways as things go on, but that is the analysis we've laid out and the way we've set forward with it. So I don't necessarily see that here, and I think that's a critical part of it. And that analysis also tells you what your focus was. You know, our focus was TEFs for the dioxin stuff. Our focus is priorities with the data we're looking at. And so we know what we're addressing and trying to provide inference on. And here I hear a lot of inferences, and I'd just like to hear what's the first one, the priority, the main inference that you want to address, so I can look at the designs of what's being done and have some feel for whether it's a good design or not. That also plays into the question of validation and feeling that you've done something reasonable, because again, your analysis strategy lays out what you're going to do to prove what you think you saw. And so it all links together. Like Abby said, it's not an independent structure.
I want to echo some of the comments I made yesterday and also refer to Chris's comments. I don't want to say amen to Chris like Abby did, but I certainly share some of the frustration that he expressed earlier. There was a lot of talk yesterday about generating more data to solve unspecified problems, and I do think we need to be more strategic about it, given limitations in resources. Part of the confusion, I think, has to do with time scale, and we'll get to that in the questions of sort of short term, medium term, long term. But I'm not sure that we could all agree on one set of goals for short term, medium term, long term. You know, we say a journey of 1,000 miles begins with a single step. And that's true in a way, but before you even take a step, you have to know what direction you're going in, right? So if you want to set priorities in the short term, that's fine. But setting priorities for animal testing wasn't necessarily the vision of the NRC committee. The NRC committee on toxicity testing for the 21st century wants to usher in a new paradigm, not to replace animal testing, but to give us a better paradigm. And it's the first step in that is not to see how well the new paradigm can predict high-dose animal studies. So we can use CompTox to do all kinds of things, and I agree with Chris. Let's specify what those are, but I would also put in a plea, as I did yesterday, that if we really want to usher in the NRC vision, let's maybe think about doing some pilot projects. What's the best set of chemicals? What's the best one or two pathways to model with the best technologies that are available today? And let's do proof of principle, case studies, and get that going. So we've got a number of sort of competing priorities, but let's just be clear what we're applying CompTox to. It can be all of the above, but let's try to parse that out so we're doing it rationally.
Thanks. Lauren?
Yeah, I had asked Bill to also put me on his list, and I think I'll just jump in here. I think we do have a clash of goals here in terms of the way that we're thinking about these early steps. The toxicity testing paradigm that was laid out in the NRC report was really a much broader vision, as Marty has said. And I don't think that, and one of the things that the committee pointed out was the difficulty in moving ahead if it's done in an ad hoc way without some kind of central and actually well-funded organization at the top that would drive that longer range vision. And I think what we're seeing here is one of the problems of the limited funding that is now being given to this issue at the federal level. It's just, we can just acknowledge that. And that in order to move ahead, we really do need, I think, in terms of the longer range vision, we need to think about what strategy the federal government or have a group that begins to really think about how to develop that strategy in a coherent way across the different groups that are do have funding to move it forward. And I have to agree with Marty that if we don't have some really clear first steps to move towards proof of principle, we're going to run into the problem of dashed expectations and perhaps reducing the will to actually move forward for the broader vision. So I just want to acknowledge that. I realize we have a question at the end around expectations.
Richard and then Richard.
So I would, my comment on Chris's comments is that we're doing all of those things. So I guess I'm not sure that I have any disagreement with most of what he said. We have a clear vision. The first short to medium term is understanding how to do prioritization. It's not clear what we're prioritizing for. Okay, we'll talk about that later on. And then longer term, we need to understand how to do risk assessment. We don't know what that means to do today. But so that's research to get there. The point of his that I wanted to actually comment on was that we're looking under the lamppost. Okay, which is that's an interesting thing to worry about. That if and his his comment about the particular study they had done where they started a study where they had I'm assuming they had a very specific hypothesis. They had assays that were appropriate. They knew how to actually run the experiment, do the analysis, so they could plan all of that beforehand. I think in the Comptex area, thinking about prioritizing chemicals, whether it's for immunotoxicity or cancer or whatever else, we don't quite know. We don't know what the right assays are. We don't know exactly what the best way to approach that is. So there is, by definition, going to be some flailing around. We have to try some things, and we have to step out from underneath the light, and we're going to make some mistakes, and we're going to potentially waste some resources. But that's the only way you learn about okay, here's some new pathways that are lit up by this class of chemicals. We didn't even know that before. Okay, if you had planned your analysis prior based on everything you knew, you would miss out on that. So it is going to be messy in its early stages, and I think it's too early to talk about validating a lot of this stuff. I think we have to collectively, it's going to take, unfortunately, a little bit of time to learn what it means to do this sort of comp tox thing, and it'll look a little messy. So that was my comment. Richard?
I agree with Chris that we need to go into some mechanisms or some studies to have goals. And one goal I think that we can do, and it's a big thing right out there, is ToxCast has all this data where in vitro and in vivo data agree, and we think that's great, but there's also a bunch of data out there where it disagrees. And so can we take a subset of that and try to figure out why they don't agree and why they do agree? And that would give us some really good information on why certain studies, why this works and why it doesn't work. We need to understand that. We have a lot of that data already out there. Can we take out, and why I would say the most likely reason they don't work is that the mechanism metabolically activated. They weren't metabolically activated in an in vitro study where they were in an in vivo study. Can we go back there and prove that? That's a very simple study. It's probably very cost effective. We can go forward with that.
Thanks. So we're getting some ideas of some strategies that we can talk about. Stan, did you want to jump in?
Yeah, Stan Barone, EPA, and in the National Center for Environmental Assessment. I want to come back to a point Abby was making about involving risk assessment earlier in the process, and I do think it's an important aspect to this, because if you're just developing a screening battery just for screening and prioritization, and you're not thinking about dosimetry, I think you miss a lot. And I come back to the point Richard was making too about metabolism. Dosimetry really can help you in a lot of ways look at the strengths and weaknesses of your different test systems, and it's something we've been talking about in the computational toxicology program quite a bit. Does your system have serum? Does your system, what kind of adhesion molecules are you using in your system? Does your chemical bind to the plate or not? What is actually being delivered to the cells? How does this compare to the in vivo dose? I mean, we have the challenges in risk assessment now of dosimetry and what's being delivered to the animal and how does that relate to in vivo doses. I think we also, because of computational toxicology methods, because of the in vitro testing, we can get at some of these dosimetry issues actually quicker and cheaper than doing it PK in whole animals. And it's something we need to consider earlier in the testing, in the comp tox testing, rather than, oh gee, now we've got all this data, what do we do with it? And some of these issues of, well, why didn't this in vitro test predict the in vivo, may be a simple dosimetry issue. Well, we don't have the dose. So I think we can rule out some of the issues if we consider dosimetry earlier and think about reverse dosimetry, as we've been talking about what's in the dish, how does that relate back to in vivo doses. So I would want to engage the risk assessors earlier in the overall paradigm shift, rather than here's all this data and now what are you going to do with it, risk assessor and regulator. Thank you.
Thanks, Dan. Carolyn, and then we're going to move on to our next question.
Okay. I just had two comments. One is just, I think, probably a very naive question, and I've been I'm fairly new to this whole in vitro assay initiative at EPA, et cetera, but do we even know what questions to ask that will enable us to assess risk? I'm not sure that question has really even come up. It's sort of like, what can we do, and let's test it and see what happens with it. But I'm not sure we're asking all the questions, and the one that keeps coming to my mind that hasn't really come up is, how do we get at the issue? So it seems to me that a lot of the historical risk assessment or toxicity data has been high dose, short-term exposure. And are we thinking in this process, as we rethink how to assess risk, what happens with these very low doses, exposures during development that don't manifest themselves until the organism becomes an adult? And I don't know how to answer that with an in vitro assay. And I think that's going to be a really important issue in terms of thinking about toxicity. And I hope that that's sort of on the table at some point. And the other issue, so one of the things that motivated me when we were starting CTD was thinking about mechanism. And so I was looking, at what resources were available 8 years ago, and looking at toxicity data available through EPA, and I could never find mention of a gene. And I thought, well, this is really weird. Then I started going to SOT meetings a while back and CTAC, and I thought, these people have never heard of genes. And that has changed dramatically in the last, 8 years or so. But I'm sort of gratified to hear all of this discussion of how do we translate these in vitro assays into thinking about networks and mechanisms. And because I've always sort of had this hunch as I've developed CTD, because those are the questions in terms of research questions that I'm personally interested in. But I'm curious how, from the risk assessor perspective, you will take that data and how you think that mechanistic data will help you in assessing risk.
So let me just react to a couple of the comments. I think it's probably a nice transition into some of the discussion that we're going to have about hazard identification. But I think one of the things that we did a few years ago, and Rita mentioned this, is that we talked about trying to understand mode of action. And that's separate from mechanism of action, in the sense that we didn't need to know all of the details. In fact, we didn't know the basis on from the standpoint of a gene or a series of genes, but we did have an understanding of what pathways were involved, or what types of activities were elicited in order to bring about this kind of a response. And we have some pretty good examples of that, where we still don't know the absolute genetic basis for this. So that was a decision that was made early on, that we didn't have to have all of the details in order to move a mode of action approach. Ideally, we'd like to know more. The other question, I think, is really around this idea of, are we starting from first biological principles to try to understand toxicity? Or are we dealing with an observational science that is much more kill them and count them. I mean, to be crass, that is the way toxicology developed. It was a testing approach, and it served us well for things that were likely to have a big and obvious impact, and things that made sense to protect the public from at all costs. On the other hand, some of the subtle toxicants that we start to deal with now that might have impact more generally across the population, might shift population susceptibilities and other sorts of things like that, that's a very different question that we're trying to ask than we approached with toxicology in the past. So again, I think this gets at how do we design the hypotheses or the questions that we want to apply these new approaches to. And so, you know, I think we're moving in the right direction with that. So I think I got everybody that had their up. Okay, Dan and then Susan, and then we'll move on to the next topic.
Okay, thanks, Bill. I'll be quick. I think there are 2 points that might be a little bit narrower, but fit into a lot of the themes that have come up here. One is just what are the test substances that we're using in CompTox now for sort of proof of principle of this broader thing? A lot of the work seems to be focused on pesticides and pharmaceuticals. I'm worried that if ultimately we try to use what's being developed now to evaluate industrial chemicals, it may not be applicable. There may be some bias from the focus on pesticides and pharmaceuticals that would have to be maintained. So that's a potential weakness that, you know, focusing on the data sets that are richest now may sort of limit the application of the tools. Second is the kickoff statements to the workshop yesterday. I heard this a couple times saying that the overall goal with computational toxicology is to be more predictive. But I think we're not entirely clear about predictive of what, because I think some of this work is sort of now seems to be geared towards being predictive of what we'd see in vivo assays in laboratory animals. I hope that our ultimate goal is to be more predictive of human response rather than the laboratory animals. I think that's really what we need to fulfill the vision.
Yes, as someone who doesn't work in this field, but who does a lot of work that uses many of the tools that people in this room are interested in applying, I would say that one of the major strengths I see here is the fact that to reach many of the goals that I've heard articulated, the people in this room and the people in the field do not have to reinvent the wheel. Many of the tools, study designs already exist. I think it's an area where you can greatly benefit the science and the impact on public health by simply adopting platforms and strategies that are used in other arenas. And I would go back to something that one of the mathematicians that does a lot of our data analysis said to me about 10 years ago. We were grappling with analytical issues in genomics data and proteomics data, mass spec platforms, microarray platforms. And this guy who's worked as part of our team for many years said, well, it's not that big a deal, it's just data. And so I think that in a lot of ways that what we're talking about here is it's not that big a deal. We just need to build the right teams to do this kind of work. And I think to answer the questions that have been posed around this table, one way to do that is simply to put together the right teams of people and get them talking. Because I really don't think that there are a lot of wheels here that need to be reinvented. They need to be just put on the right vehicle, and we need to drive that vehicle down the road.
Thanks, Susan. This is something that came up yesterday in terms of the importance of multidisciplinary and interdisciplinary approaches to solving some of these problems. And I think you've just articulated that well in terms of trying to think about how we might approach some of these things. So this is actually a very good segue into the next question that's on our list, and it has to do with applications of the technologies and whether or not there are short-term, medium-term, or long-term approaches that we might want to take to a number of the issues that are raised here. The first of which is identification of environmental stressors that may impact human health. So this so-called hazard ID approach, maybe it would be most effective if we tried to address this from the standpoint of some of the near-term activities that we might take related to those sorts of things. I think we all agree there has to be a longer-term strategy, but a good segue into this was just reiterated with the comments that Dan made about what are we trying to be predictive of in terms of hazard identification. Is it an approach that we ought to be taking that says we want to develop hypotheses to test the identification of a hazard based on our ability to analyze animal data that's already out there, or are we really going back to say, let's look at some of these strategies with regard to human risk or identifying human issues? We heard yesterday discussions about the inability to pick up Parkinson's and other sorts of things like that. Should we be looking for some of those sorts of approaches? Thoughts on that? George?
Well, in the context of computational toxicology, I think what we're trying to do with computation is to take advantage of our ability to analyze really complex data sets. And those data sets, the streams of data that we've been getting are by and large either high information content like microarrays or other similar things, or high throughput assays that we heard a lot about yesterday. Now, the thing that both of those have in common is that rather than looking at the end result that we typically do in our animal studies, so we look for some sort of disease or adverse outcome. These technologies, by their nature, are looking at the beginning of the process, not the end of the process. So they're looking at aspects of or at changes at fundamental levels of biological organization, some of which are related to those adverse outcomes. And so, the difference is in the traditional studies, we're already dealing with the adverse outcomes. In the new data sets, we're using a lot of computational power to try and analyze what's happening early on, only some of which is relevant to the disease state. So, I think we've made an incredible amount of progress actually in studies that say in genomics have been phenotypically anchored to the disease state in identifying patterns of gene expression that are relevant to different disease states. This has been, at least in the genomics area, particularly in the pharmaceuticals industry, and to some extent elsewhere, has been the whole thrust of toxicogenomics, is this identification of gene expression signatures that are relevant for particular mechanisms of action that are then anchored to disease. I think that we're really right at the cusp here of having enough information to change the paradigm a little bit. We've done the phenotypic anchoring. We know to some extent which changes in gene expression and it's also important to identify the levels of gene expression that are relevant, that are associated with particular disease states. In that sense, all that we've done is recapitulate what the animal studies do. But we've also created a tremendous platform of mechanistic information that we did not have before. We've accelerated our understanding of mode of action in ways that would have taken us decades with previous traditional techniques. I think that we're also on the cusp of starting to take that mechanistic information and, again, generate hypotheses, not make conclusions, but generate hypotheses about other disease states that might be related to these changes in gene expression for which we don't have good animal models. And so these are the kinds of things that Carolyn brought up, like latent manifestations. I mean, those latent manifestations are preceded by something. I mean, there is an immediate response. It's just not something that we've been evaluating in our animal studies. The organism just doesn't go back to the baseline and then, you know, by magic, something happens 50 years later. So I think that we're, you know, still on the cusp of doing these things. And I think what is going to be important in driving forward for you know, purposes of this group are two things. One is the acceptance of hypothesis-generated toxicology rather than checkbox toxicology, so that we can actually follow some of these things up. And the second is real attention to dose response, because I think it matters not just what's being changed in expression, but the magnitude of change and where and when during the organism's lifespan. But I think that we've got some tremendous opportunities. I didn't talk much in my example about the high throughput assays, but I think it's more of the same. I mean, it's just a different way of attacking this understanding at a basic level of biological organization. I think that doing the phenotypic anchoring in that case is more complex. I think that it's going to be informed to some extent by the toxicogenomics and other experiments that have been going on in that field for the past 7 or 8 years, which is as long as it's existed.
Chris?
Well, I had some of my own comments, but first I'm going to follow up on George's. George, I'm at a loss. I'm sitting here trying to think of a single phenotypically anchored, based on genomic response, toxic endpoint that we would say, yep, that's it. You said you think there's one now, and I can't think of it. So what are you thinking of?
Well, I think we use these all the time to evaluate whether something has the potential to be a hepatotoxicant. I mean, those are in use in pharmaceutical companies for predictive screening right now.
But there's a difference between a pharmaceutical company predictive screening exercise and a government screening exercise in terms of the degree to which you accept false positives and false negatives. We already know that. So I'm asking from the point of view of a public decision about public health, is there one? I'm curious. I'm really struggling for it.
I don't know that there is. What I'm saying, Chris, is that we have the potential to do these kinds of things. Now, I think that to you know, get things to the level of common usage in regulatory decision making, there's got to be more in the way of validation and standardization of approaches and those sorts of things. So what I'm talking about is where the science is at, you know, what we have the potential to do now, and what's actually in use in decision making in some sort of context.
Chris, I think just to sort of mediate that conversation a little bit, one of the decisions that EPA has to make has to do with new chemical substances. And at the level of an understanding of whether we should allow something to go on the market or whether we should request additional data, getting the kind of picture with regard to a potential hepatotoxicant that George is talking about would be very useful in that decision making. So that's a decision that could be made, and you can debate whether or not that decision has a big public health impact, but it could. And so, I think that's an example of an area where very early on we can start to use some of these kinds of approaches.
Perfect segue to what I was going to say, Bill. There are indeed two different types of public health decisions being dealt with here. One is new toxicants that would enter into the environment, and there's a whole process by which you go through doing that. And I agree with you, Bill, that in that case, in fact, historically, mechanistic type toxicological information has played an important role in making those decisions. Many times quite in the other direction in terms of suggesting that this is not a threat to humans when you see toxicant activity in animals, but that's the nature of the beast, of course, because the registrant is the one who's bringing forth the information and trying to make a case about safety or not. And so that is historically the way in which a lot of this information is entered into the regulatory decision-making process. But the real problem will be for the compounds that are already in commerce. I guarantee you that George would get very angry if I got up and said that every chemical that activates the NF-kappa B pathway is a toxicant. He'd justifiably tell me I was off the wall. And there are many reasons for that, part of it being that these simple signatures that we think we'd like to find are short-term, very simple signatures, and we're not sure of what their overall meaning will be. And so when we talk about improving predictability and whether we're going to predict humans or predict animals, again, I think we have to ask ourselves, what information do we have to objectively address this particular question? Now, if we look at known human carcinogens, probably the biggest group of known human, well, no, probably not. We could go for known human skin toxicants. That's an easier particular target maybe, or some of that. But if we chose a target in humans for which we had very good objective evidence of a phenotypic response, then we have something we can validate against. But until we have that, the standard has been the animal tox testing. And, one way in which to move the field forward is to try to replace or speed up the animal tox testing, in which case you'd want to predict the animal tox. Then you start working on how do you improve that. So that's one thing to be said. The question is finding solid objective evidence on which to validate. The other issue is the question of can we actually do human studies? We're talking about thousands of compounds in the environment that humans are already exposed to. If we were going to try to do genomic signatures for those toxicities from those chemicals, what we're talking about doing right now is exposing cells in a petri dish and looking for response. Wouldn't it be better to take buccal cells from humans who are working in an occupational setting with the same thing and look to see if their gene expression changes there? Are we thinking outside of that box to look at those questions? Does everything to improve toxicology and bring better information into it have to be high throughput screening? I think there are some ways we could rethink even that paradigm, and there might be resources we want to place in those settings. I think there are data sets, as we demonstrate in our paper, genetic data sets out there that tell us something about disease and human health. And I think those data also tell us something about targets for human disease from chemical exposures. And I think we haven't explored that as much as we could. So I think there's some avenues.
Thanks. Bill and Alex, and then we're going to take about a 10 or 15 minute break, give us a chance to have some smaller conversations around a cup of coffee, and then we'll come back and start this again.
Bill?
Okay, yeah, I'd like to make 2 quick points. The first point that you started is building on your comment about the new toxicants and the sort of the PMN program. And one of the things I mean, and I thought, I agree, I mean, the syllogism when I worked there was, is this, based on the structure, might this thing be neurotoxic, you know? And if so, do we have enough basis to ask for testing? And the answer is always no, or almost always no. But sometimes we're quite suspicious, but we didn't have enough to trigger an in vivo testing. And what I would like to proffer here is that in the short term, prospectively, but that maybe that should now serve as a trigger to go into some kind of testing that's relatively cheaper, or to go on the list, so that prospectively you're getting some little piece of information, and maybe a lot of little pieces of information, on those chemical structures as they move through. I mean, now it's just a possibility. That's just a possibility. On the existing toxicants, I would like to say that, you know, and this kind of goes back to the part of the origin of the future toxicity testing program is, there's a problem here, guys. You know, we have a tremendous lack of information on a whole lot of chemicals that we need to make that we're asked to make decisions on all the time. And so I guess my response to the existing toxicants is the point that was raised earlier, which is, give us something. You've got all of this information that can help us guide thousands of decisions we need to make on things. A quick example would be weathered toxaphene. There's a real issue going on now in Region 4 about whether we have an iris assessment on toxaphene, but what people really encounter is weathered toxaphene. And so is weathered toxaphene toxicologically equivalent to the parent? So that's okay. So those are my thanks.
Alex?
I'd like to share what I would describe as my confusion over what we're discussing here. Granted that Chris doesn't believe everything he said, which probably allows me to say what I believe myself. And my confusion is that this workshop is advertised as computational toxicology, and I should add, just thinking about what we discussed, computational chemical toxicology. I don't think we're discussing much of computations, and we're definitely not discussing chemicals. And so from my data explorer viewpoint, what we want is to evaluate health or environmental risks associated with chemicals. And chemicals can be characterized or should be characterized explicitly. And chemicals are characterized with, and I use data mining lingo, attributes. Some of those attributes can be computed. Some of the attributes can be measured relatively easily. Some of the attributes are what we call target attributes, something that we can't easily measure. And the goal is to evaluate what is hard to measure, what is costly to measure using the cheapest, fastest, and least invasive as far as animal testing ways. Arguably, computations are the least invasive and the fastest way, which is why we're interested in exploring this discipline. So as far as what type of data we collect, and I think what we're discussing most of the time is what type of data we should be collecting or what type of experiments we should be trusting. And it's a little bit not the same as exploring the large data space that we have already accumulated. Programs such as ToxCast excite me because it's a collection of a lot of data about a lot of chemicals. What we keep hearing is life is not simple. Similar chemicals, very similar chemicals, behave differently. The same chemical produces different effects in different species. What this tells me is that we need a lot of data on a lot of chemicals, and that's what this type of program addresses. What should we do next? We're exploring this data, trying to understand which of the attributes that have been collected in connection with attributes that we could compute are important in the context of specific in vivo endpoints. So which points are most important or less important? That's the type of knowledge that we call domain knowledge. That's what we expect to hear from domain specialists in toxicology. Our goal is to answer questions and find unknown trends in data. And so, I'd like us to discuss what type of trends we want to extract, what kind of attributes of chemicals we should be most interested in predicting, what kind we, may want to discover. And the discoveries will come out of data exploration as new discoveries come with respect to, say, side effects of drugs that nobody anticipated, or drug reprofiling. And so, that I really, as a computational chemical toxicologist now, that's the type of discussion that would be very helpful in the context of the data that we already have.
So, Alex, I think that is a piece of the puzzle that we need to work on. And you and I talked yesterday about the importance of understanding chemical attributes like methylation or intercalation or other sorts of things that are clearly inherent to the chemical structure. But what we've been talking about today is not only the issues around the chemical structure, but how that interacts with a changing biology that leads to those toxic responses. And I think this is going to be a good segue into some of the other discussions that we're going to have this morning. And I don't think we can neglect the chemicentric view. On the other hand, I think we need to make sure we put that in perspective, because that's been the challenge to acceptance of QSAR approaches in the past. And so that's something that just from experience we have to work our way through. Okay, well, we're going to continue our discussion session here. We've been working on this idea of short, medium, and long-term activities that we might undertake related to a series of topics. And we talked about identification of stressors. I think some of the comments that were made was that, perhaps we need to test this new paradigm, and we need to do it on some things that perhaps showed differences in vitro and in vivo studies as a particular example, and to take on some of these tougher challenges for trying to identify hazards that we have difficulty with our current and traditional approaches. So we agreed to let Tina start us off, and we'll continue on this for a few minutes, and then Chris, I think I'd like to have a little discussion around dose response assessment, and maybe we can ask you to start that discussion off. Tina?
I put my card up, I think, when Chris was talking, because I wanted to respond to some of that. But I do think we have to think, I'm not sure we've talked enough about short-term, medium-term, long-term. Obviously, when I was talking this morning about the culture clash, I was really making a pitch for let's keep the long-term vision in mind, and not get so caught up in our history that we don't fulfill the true promise of what this has to offer in the long term. In the short term, I think there are already a lot of things that fall into this category, and this category is broad. So you have people that are talking about data mining, but there are also people that are doing high throughput, are talking about doing studies in different animals. And I think it is a little bit like the elephant. Somebody makes a criticism of one part and it may not apply to all the parts. But, so I think in the short term, we certainly use QSAR in the toxics program. We use it in the pesticides program for things that we're data poor in. Pesticides program is generally considered to be data rich, but we don't have a lot of data on inerts. We don't have a lot of data on metabolites. We're now using it in the endocrine screening program. So I think there are short-term things that we're doing right now in terms of hazard ID, where we're there. There may be some gaps that we haven't figured out yet, but we are certainly using these techniques. But I think one of the things you said, Chris, was, should we be looking at these and validating against the animal data or validating against human data? And we need to sort of think about medium term more than I think we've been looking at. I think that's what Bill was talking about, finding something that can help us, in the medium term, answer some questions. And one of the things that I know we're faced with a lot is every other day there's an epi study that comes out that links a chemical with some health effect in people, and the animal data may show nothing about that. We have a very major epi study that NIEHS is part of called the Ag Health Study. NCI is finding associations between some pesticides and cancer, and there's almost a negative correlation between the studies that are coming out positive in the Ag Health and things that come out positive in the bioassay. And, we need more information to help us figure out, are these correlations, real? Is the animal data weak? Is there a way we need to improve it? And I think that there are some, that we have to start thinking about building sort of medium-term projects that will help us answer some of these regulatory questions. And I think that may be where the hazard ID gap is right now for the regulator.
I think that that also, again, hints at some strategies that we might take. Yvonne talked about the idea of the Tox 21 consortium essentially as being a place where some of these strategies could be talked about, and a game plan could be formulated, and maybe even some strategies for getting some funding for these sorts of activities coming out of multiple agencies could be dealt with. Tina, your suggestion, though, of focusing on some of these human studies or epidemiology reports that are coming out and really asking whether there's a way that we could apply some of these approaches to those kinds of questions, I think is a very good one. And Chris, maybe that's an opportunity for the National Toxicology Program to begin to put some priorities together with regard to some studies of that type. So just, you know, those are some things that are out there for agencies to think about.
Yeah, the other thing is, Alex said something that I think was also reminiscent of something that Bob Cavlock said yesterday, and that has to do with, I mean, for example, we need to have the toxicologists sort of have to figure out all the tox pathways so then the data can be mined. And I think the toxicologists are kind of hoping that some of the data mining exercises will help guide some of the more basic research on tox pathways too. So I think that there are sort of conflicting expectations there, and that may also be something we need to sort of work on in the medium term.
So yeah, I'd like to second that, and I think that's, you know, is an opportunity, you know, on top of all the challenges that were outlined, where again, we do need to foster these collaborative groups and multi and interdisciplinary groups, and again, not only within the government, but also bring in the academia, as again, you know, EPA is, you know, doing that by funding computational toxicology centers, and, you know, I'd like to disclose that I'm associated with 2 of those, and I think that really is allowing the field to go forward with marrying the experimentalists and the computational people in academia with trying to think about what the agencies need. And this is not the usual philosophy of NIH funding, where NIH doesn't want to dictate and neither EPA wants to dictate what the science should be, but there is a lot of benefit in trying to tailor the basic science to the applications and have a shorter path to translation, if not to human health, at least to regulation or practical applications. So I think toxicology is a great example of very applied science, and there's, as far as I'm concerned, nothing bad in that definition.
Good comments. Alex, and then Chris, maybe you can sort of lead us into the next one. Okay? Okay. Chris?
So I think medium-term goals is a good idea. Short-term goals is a good idea. Long-term goals is a great idea. Make them public. Make them clear. Lay down the path by which you intend to go to them. I think that's extremely important in doing the things that Ivan wants, and that is to gain funding for the area, to gain acceptance of the area. I mean, you want to have people understand what you're trying to achieve very clearly, very concisely. I'm here with 2 agencies, not just one. The National Toxicology Program is one, but NIH and NIEHS is indeed a completely different agency. And there are a lot of areas related to this topic that NIEHS is pursuing. The Ag Health study is certainly not sitting in isolation. There will be a number of grants that will be proposed to follow up on some of those pesticide findings from the Ag Health study, and of course those will then be funded through our extramural program. A lot of the questions we have about susceptible populations and how do we screen susceptible populations, NIH's roadmap activity on chemical screening, small molecule screening for biological targets, which is what NCGC is actually there for, is a huge effort that's contributing a tremendous amount to understanding how to use different types of cell lines, how to put them through these types of assays. So I think there's a number of activities. What we have to understand is we need to, when we talk about it in a public health context, we want to be clear in this public health decision context what we mean by research that's medium term, long term, and research that's really short term and has aims directly at public health. The question of dose response, shall I go into that now, Bill? So I've made my entire career around analyzing dose response for the purposes of risk assessment, and certainly I'm not going to step aside from that just because we're going to start doing high throughput screening or other issues. I think it's an incredibly important part of this equation. I've always been a firm advocate of using the actual quantitative response. In fact, Ruben Thomas is here somewhere, and we have a research project right now that he put at SLT, at the annual meeting of the SLT, where instead of trying to predict yes, no for cancer, which we think is a waste of time, we do have strong feelings on some of these things, we're actually trying to predict the slope of the dose response curve. To us, that's a more reasonable thing to predict. And then we're trying to predict when that slope is significantly different from zero. Then, if you really want to interpret that as cancer, yes, no, you can. But what happens then is you gain strength in the overall analysis, because instead of trying to predict a 0-1 endpoint, you're predicting a continuous endpoint, and small subtle differences can strengthen the overall analysis. So I'm a firm believer in using dose response in this. The high throughput screening activity that we have, as Chris told you yesterday, it's 15-point dose response curve, over 7.5 orders of magnitude. Of course we believe in dose response being important in this attribute. In this issue, what I'm suggesting is that there are two levels to using that dose response or interpreting that information in the context of using this in public health decisions. The first is using that dose response information like we do the animal dose response information for carcinogenicity studies to make a decision of yes or no. Is this a carcinogen or is it not a carcinogen? That's the first stage. And you really do need dose response to do that. I'm a firm believer. Then you go to the second stage. Is this now predictive of something else? And in this case, it's not the same as the animal case. In the animal case, we can use the same experiment. We can use the same information. At least we've historically accepted the information. George might challenge me on can, but we've at least historically accepted the information that the dose response from the animal can be used to make some statement about safety in humans. I think we have to prove that in this case. And that's the difference. That's why I hesitate to say I'm going to jump on the dose response issue right away. I can see the dose response issue as telling me something about yes, no. It's harder for me to see the dose response telling me about the entire pattern of response over a continuous dosing range that's relevant to an in vivo situation. I think that's something we have to work towards proving. It's a second level of the overall analysis.
So, Chris, let me just explore a little bit of that, because I think one of the issues that is intriguing about high throughput screening and a number of the approaches that we heard yesterday was that you can look at a much larger dose response. You mentioned 7.5 orders of magnitude. I mean, we know that with so many of our in vivo studies, we're really restricted to typically an order of magnitude of dose response simply because of the numbers of animals that we're dealing with. And in a few cases, we might have gotten to 1 or 2 orders of magnitude if we had a very potent biological activity. So I think that there's also some real benefit in terms of, call it almost range finding studies that would allow you to better design animal studies that would take advantage of the limited numbers of animals that you do have available and target your testing.
I fully agree, Bill. I think there's information. as we've shown, you've shown with papers, I've shown with papers, there's information and mechanistic data to give us some indication of what the dose response will look like. In the case of the data we're looking at, after having looked at 11 different cell lines, 1,500 compounds, all this dose response from the high throughput screening for 17, 18 different assays, what we see is a great deal of variation from one cell line to the next, from one assay to the next, in terms of the consistency and the shapes of the responses. We've got responses that go up and down. We got responses that go down and up. We got responses that level out. All of this needs to be worked on in terms of interpreting it for the dose response issue. I think it's going to be a little more clear for the yes/no issue, a little more difficult for the dose response because of the nature of the data and the magnitude of the information we're going to be getting back. With 300 different assays showing different dose responses for a single chemical, understanding how we're going to use that information to make a final statement about dose response in humans or even in my next test animal is a research issue to me.
So even if you see differences in terms of the absolute magnitude of the range of dose response, the shape of the dose response may be consistent or wildly inconsistent, and that's going to be useful information for you to understand as you move into a higher order type of a test. Absolutely. All right. So Yvonne and then George and others, put your tents up.
So Chris, I think that collecting dose response data in high throughput, high content assays should be a must if it is possible. And again, there are different types of assays that can or cannot contribute or are amenable to multiple doses. For things that NCGC does, I think that if they can collect 15 doses and they can do that before Chris has his morning coffee, amen to that. I do agree with you that it introduces a tremendous additional level of complexity because you have different cell lines, you have weird dose response shapes, and sometimes you don't know what to do with that. However, if you look at it from the data analysis standpoint, what you're looking for, you look for matrix that has data that doesn't have a yes or no response, where most of the responses in most of the assays for most of the chemicals thus far are no response. So we had a bit of a discussion yesterday on, if we screen more, are we going to enrich the matrix, or are we going to get a bigger sparse matrix, and how do we pick the assays that give us some information if we did invest the money into running this assay on these particular chemicals. Because whatever information you have, we have to think about how to use it. Right now, we're feeling our way around again, and we're trying to use active, non-active. Then hopefully we'll at some point, or for some of the analysis, we'll get to the IC50, EC50, some other metric. Maybe when we know what types of analysis we're comfortable with and what paradigms we're trying to follow, and there's enough computational power, we can dump all this data in. Because the ultimate goal is to find something that maybe you don't know why it's correlating, but it's correlating strongly, and having enough data and enough endpoints and being statistically rigorous about external validation of these results, then you're looking for as much data as you can. So if you can collect dose response, even though we may not know what to do with it right now, I think we should do it early on.
It's nice to have something that we can all violently agree is important. So I wanted to talk about two separate issues around dose response. One is I'm one of those people that believes that you can't separate hazard from dose in defining whether something is toxic or not. And I think one of the limitations that we've had in large database analysis has been the artificial dichotomization of tox data as something is or is not a carcinogen, something is or is not a developmental toxicant. I mean, really, something is or is not a developmental toxicant at a certain dose and at a, when dosed at a certain time kind of thing. And that granularity of information is something that we have somehow artificially divorced. We understand what we're doing in our own little field, but people who go and analyze these data often don't. And so, if we can find ways to incorporate potency information, dose information into these assay systems, so much the better. One of the real the second point that I wanted to make is some of the things that have been, I think, real advances in dose response analysis that I think are relevant to a public health context are identifying what happens at and below the no observed effect level from traditional animal studies. And we do have some examples of that from gene expression analyses. So, one of the arguments that we've had that can start to be settled by some of these statements is whether we're not seeing things below that no effect level because we don't have the statistical resolving power, which we don't, or whether there are real dose-dependent transitions that happen. And so some of the experiments that are being done in the toxicogenomics area, for example, are in fact getting at this issue of dose-dependent transitions. So, are there real qualitative differences in the expression of genes which have a different resolving power than just the ultimate adverse outcome at lower doses where we also don't see pathology, that sort of thing. And so there's a good paper on that from Rusty Thomas's group that was published last year. We've published data over 5 or 6 orders of magnitude dosing in developmental tox in terms of development of the reproductive system and its sensitivity to estrogens that can be analyzed for dose-dependent transition and also the mechanisms of action. And I think Yvonne's group has actually taken some of our data and really made great use of it in that sense. So I think that those are the other aspects of computational toxicology that I think are really going to be informative for dose response assessment. And I think that they get at Stan's question or Stan's comment from earlier, which is, there are a lot of things that the regulators, the risk assessors could really use in dose response assessment that, given the right approaches, we might be able to address.
Thanks, George. Lauren, Sarah, and then Tom.
Yeah, I guess going a little further with the points that George has raised. I guess first off, I guess I see toxicity a little bit different maybe from the environmental regulatory side in that I think everything's toxic. The question is at what dose. And so, yeah. And so I think with these kind of data, we have the opportunity actually I think we think about various uses of these data. One is sort of in a green chemistry context in terms of, does it make sense to substitute this chemical in for another chemical? And the other is the much more detailed dose response information that is brought to bear on things that are out in the environment, which are potentially very costly in terms of regulation. So I think just in terms of the green chemistry side, in a sense, we don't need as much detail on exactly what are the specific outcomes that we might predict. We might look at a series of indicators of basic biologic activity and link these to specific doses and compare across similar chemicals with this spectrum of biologic activity in terms of alternatives analysis for green chemistry. And I think in a way, I wouldn't be surprised if this is something that is done commercially. Certainly the regulatory agencies aren't doing that. But with this whole issue of green chemistry and providing some general public knowledge, I think it's critical as we advance this field forward that we actually provide the public with some tools that they can use to make these kinds of decisions and provide some strategies for thinking about what do we mean by fundamental biologic activities. There's a very recent paper by Simmons, and I think one of the authors is here, looking at different stress pathways. And so there is this other use. I do want to bring up this issue that came out in a national NAS report around background exposures. And I think we had a lot of discussion yesterday, kind of wringing of hands about, well, how do you take into account things like nutritional status, background exposures to other agents, and so forth. And clearly, in terms of interpreting, looking at how we use the outcomes from these various experiments and how they apply to the human condition, this whole issue of background exposure is something that I think we need to work through very carefully. And these data can give us a way of exploring those issues, these kinds of tests. But I think we have a long way to go in taking these things and interpreting them in terms of the human case. And that we do really need to think about the issue of additivity to background. So just looking at a result from a study and seeing that, oh, if I look at these concentrations and use some kind of pharmacokinetics to interpret them back to the human case, that I've kind of addressed this issue, I think we have to be careful that we don't take that approach, that there is this whole issue of background additivity for both carcinogens and non-carcinogens, that we can use these kinds of approaches to explore, but we need to be very careful in terms of using them in a regulatory context.
Thanks. That obviously that's a topic that I've had a lot of interest in, and it's good that we think about that. I think the discussion that we heard earlier from Chris about the benefits of really understanding a large range of dose response really helps us in that, in terms of putting some of those things in perspective. Sarah will be next, and then I have Bruce, Richard, and Shukmei.
As important as dose response is, of course, it exists in a framework of comparability of tests, and we can't lose sight of the fact that our exposures can be very different depending on the test that we do. And we ought to be thinking about how comparable these doses are as we compare the results of different tests.
Thanks. Good caution. Bruce?
Okay, I just have one comment about before we leave dose response, and then a placeholder. The comment is dose response to whom? And I think what we can do prospectively is take data from SNPs analysis and molecular docking data for chemicals of interest, looking at particular genes of interest. And you could engineer those genes for those SNPs into some of the cells, let's say, that NCGC uses. So you could begin to generate data on and test whether or not the predictions from the molecular docking would be hold water or hold metabolism. And that should help in terms of the risk assessment. I'm trying to fine-tune the risk assessment. It's one thing, we all believe in dose response, sure, but it's the dose of something to you and a dose of something to me is maybe very different in terms of the consequences. The placeholder is that I'd like to see before we break up is that we've talked about you, as in all you all, should do this and should do that going down the road. I'd like to see if we can identify some entity, and whether it's the National Academy or somebody else, who would take responsibility or be willing to take responsibility for beginning to pull this together so we aren't having the same conversation 5 years from now, that in fact we identified some priorities, we addressed some of the issues, we run right at the issues that Chris raised, I'll say it like that.
So just to address that last point, this is not a consensus workshop, and so we're not going to reach those kinds of conclusions, but it is an opportunity for us to really air some of these kinds of issues. And I think I'll put words in Chris's mouth. I like doing that, actually. NIEHS has funded this effort for exactly the reasons of getting these kinds of issues out on the table and initiating this dialogue about how we can actually move some of these things forward. And so I think the benefit of this is that there will be discussions at NIEHS and in the National Toxicology Program and with other collaborations of agencies that will then say, okay, now how do we move this forward?
I want to hazard some comments from being outside the field. There may be issues that I just simply don't understand or I'm not aware of, but it's interesting for me to see that how words are used. In my talk, I put up model and how model is used so differently. So here we use words like computation, we use words of prediction, and I think they mean very different things in different areas, and it's relevant to the range of talks that was given today. I think that most of the discussion I've heard today is based on statistical prediction. And so statistical prediction is based on the current data set you have and extrapolating from it either between data points or venturing outside of those data points. A principled prediction, let me just make up that word, might be based on a differential equation, which would claim to be able to go outside of measured data points in a way which might have more confidence. And in some ways might be more what we think of that we can take a compound, for example, that's completely unknown and be able to make an assessment of it without it having a close correlate to some data we already have. And again, it seems that most of what has been discussed here is statistical prediction. But some of the talks, for example, when you start talking about biochemical networks or you start talking about quantum chemistry, start informing true hello, NAS Pizza Parlor. So the person I think is still in the line is going to enjoy the rest of the discussion today. So I mean, these other forms of prediction are coming down the pike, and along with that, I've talked about functional assays in a sense when I talked about a clearance assay. And I think the funding has just been cut for the agency due to my rudeness. Sorry about that. The functional assays I talked about developing in the context of virtual tissues, that also has a computational background to it. And for our purposes right now, perhaps it's not even useful to distinguish between a computational virtual tissue and a tissue assay, a functional assay like I discussed. And we've also talked about tissue engineering, which relates both to developing tissue models, but also I think has a lot to say about tissue remodeling, which is something that happens downstream in a toxicant dose setting. So in all those contexts, I don't see a lot of discussion here about how we start about a roadmap for bringing those kinds of science to, and I'm going to call it predictive science in a principled sense, into the stream of information that toxicology deals with and makes decisions on. In some ways, it's very daunting coming into the field because the high throughput screening is so pervasive that it's hard to understand how we would actually start talking about 10,000 tissue cultures in a sense with a functional assay. But in any event, I think to the extent that these fields are coming online, that they're going to make a contribution to toxicology, we need both specifications and a roadmap for what would you want to see from a functional assay that would make you pay attention to it and would say, okay, we don't need to do this testing in an animal anymore. So in a sense, it goes back to what Chris was talking about specifications. I think if you're going to nurture these fields coming in and feeding into the stream of information and decision making in toxicology, it would be a great benefit to give guidance to those fields and say, here's the benchmark you have to start matching in order to be relevant to what we're trying to accomplish.
I think those are very good points. At the end of the discussion, we wanted to talk a little bit about managing expectations for the field, and I think some of what you're getting at, Richard, is really about that. There's been I'll call it a reaction, negative reaction maybe, on the part of some about the vision of the 21st century tox, simply because there's this view that unless we can do everything, we can do nothing. And so I think if there is, there's an opportunity as we get to the end of this discussion to talk about managing those expectations and what it will take to sort of lay out those expectations and roadmap, that'll be very useful for us. Shukmei?
Well, I want to come back to a point that Chris made early on. How would this kind of research lead us to really to advance or improve public health issues? I think we really now have a new opportunity to really take computational toxicology, not just from the bench side to the bedside, but directly go to the population and basically use a lot of the new possibilities, like for example, deep sequencing, generating lots of data from each individuals, taking human samples and analyzing. For example, last week I listened to David's talk was like all these sensors that can detect thousands of chemicals that are actually in the populations, and then we can actually use this end from this end and looking at how we can even generate those response curve from real population if we have very accurate predictive markers that have concrete cut-off points that is tied to phenotype or pathology of humans, and then using that and then combining with what we learned yesterday, in which we can actually see a lot of the high throughput screens that can generate mechanisms, and then come to kind of a midpoint in a new type of science that can actually address both public health issues and mechanisms and scientific issues. So I am very optimistic in terms of wanting to see that computational toxicology moving directly into population biology studies and utilizing like human cells and perhaps a human body fluid collections and also ambient measurements of direct measurements of thousands of chemicals using high sophisticated sensors. And that is actually what I fancy, and then from that and combine it with what we learned yesterday.
So I think that really, is the ultimate goal of all of this. But I think the question of how do we approach this and how do we bite off some bite-sized pieces that we can really make some progress on and demonstrate the value of these approaches is going to be critical for us as we move toward that. There were two other areas that we wanted to work on as we sort of work our way down the list, and they were related to comparative safety assessment. And we've already heard a little bit about this. Bill Setti mentioned the issues of aged toxaphene. Lauren talked a little bit about green chemistry, and we clearly have some experiments going on where we're using things in the environment that have not necessarily been tested as well as they could have, and perhaps we could feel more comfortable with those kinds of things going on if we had some of these kinds of comparative data. So I'm going to ask Dan Axelrod to say a few words about that. Janelle, do you want to jump in before we move into that new topic? Okay.
I was going to mention, as far as dose response, I do think that is coming from the Office of Emergency Response, Office of Emergency Management, I'm sorry, that in the dose response world, if you take a look at, the physiological versus the pharmacological effects, I think it's really critical to identify those physiological effects when we're looking at exposure guideline levels and developing those, because oftentimes we want those low-level minimal effects so we know what to recommend to the public, what they can be exposed to or not. So just in the world of computational toxicology right now, it's a golden opportunity to be looking at those low level and what the uncertainties are associated with those low level physiological effects as well.
Okay. Right. Good, thanks. Dan?
Okay, thank you. Lauren and Bill, I think we're thinking about this question the same way I was, which is reassuring before I start talking. But I don't want to sort of recapitulate any of the discussion that we've had about strengths and weaknesses of comp tox, because I think a lot of those that we've covered already sort of carry over into this topic. But I want to think about this question about comparative safety of stressors in terms of particular applications where I think comp tox could potentially help us a lot with these types of questions that Lauren and Bill were alluding to. I'm just going to throw out a couple scenarios. They're real ones. First one is where we're looking at ongoing changes in the marketplace in real time, where there's substitution going on for a widely used existing chemical that's being phased out of use. There's been several examples in recent years with brominated flame retardants, some applications of bisphenol A, some applications of phthalates. For the most part, these aren't changes that seem to be sort of slow in building. We reached a tipping point where these chemicals were being removed from the market, either because of regulatory pressures or just general concern. One specific example was pentabromodiphenyl ether, one of the PBDEs, a flame retardant that is used in polyurethane foam and furniture. This chemical was phased out at the end of 2004 and voluntarily in the U.S. following some states legislating similar actions which would have taken place a few years later. But once this happened, it happened pretty quickly. And you had a lot of furniture manufacturers, foam manufacturers out there who've been using this particular flame retardant in their products for many years. And all of a sudden, this market is in transition. And all these manufacturers have to decide what flame retardant am I going to use in its place. And of course, chemical manufacturers are giving them some guidance. But from the EPA perspective, we're looking at this as here's an opportunity to try and give information to the market to let them know what we think we know about the relative hazards of the different chemicals that might substitute. So this was a project that was done in EPA's Office of Pollution Prevention and Toxics. They identified a broad range of flame retardants that might be used in place of penta-BDE, compiled all the existing toxicity information, and a pretty heroic effort, pretty quickly put out a report that compiled all this information, put together in tables. So you had a standard table for each flame retardant. You could look and see the same information in each of these tables. But these tables were all based on the existing tox information and PChem information that was available for each chemical. And of course, that's going to be very divergent for each chemical. So this was a scenario where if you had a bunch of chemicals that you know are candidates to go into the market in the near future, and you want to give guidance to the industry on what can we say about the relative hazards and how can we avoid getting the market into another situation a few years down the road where maybe the chemical that won in the market maybe has similar problems. So if you could use high throughput screening and comp tox to do quick evaluations of that whole set of chemicals, looking at the same markers, the same endpoints for each chemical in the set, I think that's something that could potentially be very informative. And it's not just this chemical. We've seen the switch over from BPA to I don't know what chemicals took the place of BPA in plastic bottles. But I suspect that we did not have a comparable set of data for the various candidate chemicals. And so that's something there that would have been very useful too. So that's one broad area where you've got substitution for an existing chemical that's widely used and being phased out. The second scenario, I don't think we've heard nanomaterials mentioned in this workshop yet. I don't know if nanomaterials are amenable to assessment with high throughput screening, but we've got a very broad broad range of nanomaterials with very little data on each of them. We have very limited experience with in vivo testing for these materials, and even some questions about, how do we define the dose metrics for what those studies should be. And most interestingly, I think, is for a particular nanomaterial, there can be a lot of minor tweaks that you can make to give it various properties. So we don't just have carbon nanotubes, we have hundreds of possible tweaks that one could make to a carbon nanotube, and to give it different properties in its intended applications, but may also give it different toxicological properties. So I think it would be really great if it was feasible to take those hundreds of variants of carbon nanotubes and do some assays on all of them so you can get some comparative data. So those are some possible scenarios for application of CompTox that are sort of, I think, different from a lot of what's been driving the discussion up to this point.
Thanks, Dan. I think Bob Tanguay actually talked a little bit about the nanomaterials testing in the worm test yesterday, and it looks like there may be some opportunities to use some of these kinds of screening assays to really be a benefit to us as we begin to look at some of those issues. Any other comments relating to comparative safety assessment or the application of these kinds of approaches to comparative safety? Yes.
Well, I just want to point out, and I'm sure Richard could add to this, EPA has in the ToxCast queue a set of chemicals that are considered to be green chemicals that they'll be testing. And that's something that, like, I've really strongly advocated for. I work with the EPA Design for the Environment program. They're the ones that have really pioneered these alternatives assessment techniques. And, it's just such a tremendous opportunity, I think, for to use ToxCast to see, is it useful in informing what the Design for the Environment program likes to call informed substitution.
Yeah, we certainly want to do that. We don't have specific chemicals yet. We've asked for nominations. So
we've got a list.
Good.
Yeah.
All right, great. And we're also doing nanomaterials, so we'll evaluate that too.
So Richard, while you have your microphone turned on, the next issue that we were going to talk about was applications for prioritizing testing. And again, we touched on this through the course of the morning, but I'd like to sort of focus as we wrap this session up on some of the benefits that we might see to the approaches that we're taking now in terms of prioritizing for additional testing. And I'm going to ask Chris to jump in from the NTP standpoint, how that might benefit the NTP program as well.
Okay, so I have a few comments to get started. So first of all, I believe it's consensus within the EPA, OPPTS, and the other offices we work with ORD that prioritization is the short to medium term goal of computational toxicology. That will be the deliverable. And I want to very loosely paraphrase some comments that Jim Jones, the head of OPPTS, has made. He's essentially said that yes, prioritization is important. There are chemicals out there, whether they're new chemicals that Bill was talking about or existing chemicals for which we don't have any significant toxicity testing data. It would be protective of human health if we had more data. But clearly there's no, the resources are limited. You couldn't take 10,000 chemicals and run them through the standard battery of all the guideline tests. The REACH, the folks who are dealing with REACH are coming to that realization that what they have seemingly mandated is actually a lot more than maybe they had thought about. Okay, so somehow you have to do prioritization. Now, once you have prioritized chemicals, however you do that, it's not clear what you do with the guys at the top. And that's really not necessarily a science question. That's almost a policy question. Which of those chemicals, how many of those chemicals do you decide need to go through what tests? What tests do you put them through? Who pays for those tests? How you regulate based on those tests? So, those are issues that will have to be dealt with at another time. Okay, but given that prioritization is important, we want to do that. There are really two avenues that you can go down, and we've really talked about these. One is you could say, what I really want to do is have a prioritization scheme which essentially gives me results like the current animal tests. So like Alex was, what he presented, what Russ presented, those are really models where you can take the in vitro data plus the in silico data, all the chemical descriptors, and you can come up with a scheme that takes a new chemical, you put it through there, it puts it somewhere in, does it look like a carcinogen or not? Does it look like an immunotoxicant or not? You can put dose response data in there. Does it look like it will, have these sort of responses at a low dose or not? So anyway, we're trying to essentially mimic the animal data, and you could imagine going through sort of an explicit validation phase where you decide, okay, a model has to have a certain predictive power, whether it's sensitivity or specificity, before you believe it and you use it. So we could put all that into place. The alternative route, which Marty actually brought up yesterday, which is kind of the vision, the NRC vision, is that you forget about animals because animals are not humans, and really what you do is you go through and say, do these chemicals hit particular pathways that we know or we believe will be associated with toxicity? And you can do that in a dose response way. So you can say this chemical hits a particular pathway with a pseudo IC50 of 10 nanomolars, and here another one, it's 10 micromolar. Clearly the one that hits at 10 nanomolar seems to be, you'd worry about that more, so it would go up higher in the priority list. Okay, so those are really two routes, and to close, what I want to do is pose a thought experiment, which actually could be turned into a real experiment that maybe makes this a little bit more concrete. And in a sense, it's almost the simplest experiment, but nonetheless, I suspect it's very controversial. So, we all obviously worry about endocrine disruption. So there are a series of assays that you can run today that measure, and let's just talk about estrogen receptor. Okay, you can run an estrogen receptor binding assay, an agonist assay, and an antagonist assay. We could find 3 or 4 or 5 labs, each of which we asked to develop those 3 assays. We could couple them with a metabolic pre-activation assay, so we actually get all the metabolites, we put it in there, and we go through a validation phase for each of those assays. We have a certain number of positives and negatives, and we show how well those labs reproduce that set of assays. Okay, there's no huge technology leap to be able to get to that stage. Then we queue up 10,000 chemicals. As Alex points out, we go through very careful analytical chemistry and make sure that the chemicals we're asking these people to test are really, are what we think they are. And you run it through, and you get a series of, essentially for each of these assays, you get an IC50 and you get an efficacy. And so we would say chemicals that have an IC50 of 10 nanomolar in some of these assays, we obviously want to set as a higher priority than ones that don't have any activity or that have 100 micromolar activity. 2 chemicals that are both at 1 micromolar, the one that is a total agonist, we're going to prioritize above the one that's a partial agonist, so on. And then at the end of that, you've taken your 10,000 chemicals and you've ranked them somehow, and then you have to think about, okay, so what do we do? So what? There's obviously a whole series of next step assays you could run them through. So that would be, it's a very simple experiment to think about doing it. It takes into account a lot of the things we've worried about today, but I'm sure that there are big holes in that we still don't know exactly, is that quite the right experiment to do? And it doesn't matter whether it's estrogen or it's P53 or whatever else that we know something about. So that's my intro.
Thanks, Richard. I think as we consider those sorts of thought experiments, this is going to lead us into our discussion about managing expectations, because if we think about Richard's experiment and we are only 99% accurate in terms of this particular issue, we're going to basically end up with 100 or more chemicals that basically we have perhaps a false positive response, perhaps a positive response, but that's going to be an issue that people are going to want to deal with immediately. And so that's the issue of how we manage expectations and communicate about these kinds of assays. So we have to be very careful as we think about how we bring those kinds of things online and manage expectations of the results. Rita?
This is along the lines of managing expectations and prioritizing as well. And I'm having these thoughts because of older experiences with prioritizations. And generally what we've done in the past, we, the scientific community, not even necessarily EPA, is when you get a hit on some sort of screening assay and you move it to the top of your prioritization queue, what you generally do next then is run it through a battery of assays. And then the next question is, what do you do with all the positive, negative responses you get in the battery of assays? This is a pattern that I've seen repeated for 25, 30 years at this point. We as a community haven't had, I think, a clear procedure except for test rules as to what do you do next when you've got a hit. And truth to tell, the majority of us are not bound by test rules. And I do think that this might be a useful field of inquiry. All right, it's now we've got the top 100 chemicals. What are we going to do? As you pointed out, the Europeans had a nice scheme, but they're finding that there are an awful lot of chemicals that are in their top priority, and I think they're going to have to do some reconsidering as to how they're going to proceed. Before we put ourselves in the same position as a U.S. and somewhat broader scientific community, I think it might be useful to talk about these things.
Rita reminds me how old I am. The first job that I had when I came to EPA in 1979 was to design a tox testing scheme, which we published about a year or so later. But what's old is new again, I guess. Lauren and then Chris. Yep. Okay.
So actually I want to get back to something Dr. Axelrod said with the comparative safety analysis. I assume you're talking about the long term. Because I think if you try to use some of these approaches, CompTox, whatever you want to call them, at this point, I think that would be actually a bad use of it. Because if you come up, you run through and let's say none of the substitutes raise any flags. So does that then give you confidence that these are actually safe? I mean, have you done the right test? Is there even a test for some of the effects this compound would have and so on? So I think in the long term, once things have been validated, that would be a tremendous application. But I think in the short term, it would actually be counterproductive.
I agree. It's all contingent on having assays that you have confidence in, which I didn't want to repeat during my remarks. But yeah, that's clearly a premise.
Lauren?
Yeah, I think once we do have some assays that we do have some confidence in, and we think about the assays as prioritizing for testing, I wonder if we actually need to merge the two tracks, prioritization versus characterization, based on what you have coming out of a test. Because ultimately what you're doing as you're getting these signals that there's toxicity is you're developing higher and higher probabilities that there are effects. And one of the uses of this scheme is for regulatory agencies, but another use of this kind of information, again, is the broader public, the states, but also the broader public. And I wonder if there is a way of moving over time to a system where we don't just stop at prioritization and wait, which is what tends to happen. I think with the endocrine disruptor program, that actually has been the basic outcome, is now we know 10 years later, with some very useful information, what chemicals to move to, that makes sense to move along in terms of tiering. But we also have some indication that these are lighting up for certain kinds of hazards. So if there's a way of capturing that information and somehow making it available in a useful way to the public, because it can be misused as well, but making it available and used in a useful way, and thinking about different schemes for basically projecting hazards, something that it's really a rule-in kind of concept, that something that passes this particular screen that might not be endocrine disrupting, for example, doesn't mean it's safe all around, but it does mean that it's probably less probable than some of the others that are lighting up through that screen, and so it's useful to tell the public about those things that are appearing to be hazardous.
So Lauren, I guess I would just temper your comment a little bit by saying that maybe we're not developing higher probabilities that decisions that we're making are related to risk, but really that the decisions that we're making are science-based. And so that actually gives us a little bit more to offer to the public about the kinds of decisions that we're making. So we've got about just a little bit less than half an hour left, and there are three areas that we wanted to touch on. One of them, slightly out of order, is what are appropriate decision rules? And Gail, I think maybe it's starting to sort of feel like it fits right in here, and then we'll talk a little bit about the public and how to communicate some of these things and how to manage some of the expectations. And Trey, we'll ask you to jump in at that point. So Gail, could we hear some of your comments maybe?
Yeah, I've actually had a great 2 days. And I know why I was assigned this particular topic is because NIOSH doesn't have a computational toxicology program. So as far as decision rules, I've got a whole list of questions that I think that I would like to see answered before we would incorporate this, this tool into our cadre of tools, or into our toolbox. So I guess, the ultimate question is, what are we going to use computational toxicology data for? And so, ultimately we would like to use it for either, although NIOSH doesn't do regulation, but we do public health, make public health decisions, we make, standards. And, I just don't think that, we're there yet. And I think that's pretty much been the general consensus of everyone here. But, what we could use it for is hazard identification. And, something that like Bruce brought up as far as emergency response, where we, go into a place where there might be a mixture of chemicals, and it might help us narrow down to, what is the prime actor, such as, what we found with the popcorn lung. And we went in, and there was a whole mixture as far as, the butter flavorings and which chemical was responsible for the bronchiolitis obliterans, that was seen. And so, it could be useful, in that, in that situation. And I do believe that it could be also useful in substitution, because we actually run into that a lot, where, especially when methylene chloride was being phased out and they used bromopropane in some of the adhesives, and we found a lot of neurotoxicity in some of the furniture manufacturing. And I guess the bromopropane industry has a very good lobby, because when perchloroethylene was being phased out, they decided to use bromopropane in place of perchloroethylene, and so there was a lot of dry cleaners up in New Jersey that also had some neurotoxicity. So just if they would just some of these substitutions maybe could be tested ahead of time in the appropriate method. I think that would be helpful. I think prioritization would be a great way to use this technology, and this would certainly help. I think a lot of agencies that aren't flush with money to at least be able to narrow, their resources, or be able to pick what resource, their meager resources, whatever they have left, into developing those priorities. Some other questions I had is, what is the information available? And I think there's a lot of data out there, and people are putting it in databases, but I'm not really for sure how it's being translated into what would be useful for for public health. And I think Bruce had an intriguing idea, is to go backwards. We have human data, and then why don't we go back and try to develop a model of, for either a class of chemicals, or just to see if it works. I'm concerned about accuracy of data.
A database is only as good as the information that you put into it. And I think there's laboratory validation concerns, but there's also model validation concerns as well. What's the relevancy of the information that we're getting from computational toxicology, and how does that fit into the exposure to disease continuum? And is that toxic effect that's seen in these assays, how do they relate to a health concern? And then that segues into dose, because I'm an exposure assessment person, so dose is always the key to anything that I do. And so we need to be able to understand and relate what happens at a 96 well plate or a 1,000 well plate and to what happens to an individual. And then I'm also, NIOSH isn't one of those agencies that's flush with money, so I'm always concerned about cost and both in funding and in resources. And I know that it takes millions of dollars to generate to generate the data in some of these high labs, high throughput labs, just to set up the labs and whatnot. But similarly, the price tag is the same for developing a database. So with that, I think that we need partnerships and collaborations are going to be very important. And the multidisciplinary and interdisciplinary slide that someone put up yesterday I thought was so appropriate, because that's what it's going to take to fill in these gaps that we have. It's not just going to be the toxicologist or the laboratorian. It's going to take a team of people to do this. And standardization, I think, is going to be very important. One of the things that we have found at NIOSH when we're trying to put together an occupational exposure database, and industrial hygienists like to use their own metrics. And so I think that we need standardization. We need standardization within NIOSH, but when we go out on field studies about our standards, how we measure things. But I think the genomics area with the Miami plan was actually a very good thing to do, and I think computational toxicology might want to consider developing such a standardization process on their own. But I found that just after listening to the last couple of days, I think that computational toxicology is an incredibly powerful tool, and that I think that it will fit into the arsenal of public health.
Thanks, Gail. Chris, do you want to jump in at this point? Again, there may be some opportunities to talk about the issues around prioritization, as well as some of the comments that Gail just made about some of these decision rules.
Oh, I think Gail hit the decision rules on the nose there. I believe she walked us through all the things we have to think about as we decide what we're going to use this. And I'm very happy to be a partner with NIOSH through the NTP in terms of addressing some of the questions that we've always historically addressed together. I wanted to talk a little bit about priority setting. There were a couple of things that weren't said that I think are incredibly important in thinking about priority setting and what you're priority setting for. So from my perspective at NIEHS and looking at the NTP, we have two priorities we want to set. First is, what research does NIEHS need to fund to advance this area of science? And that's something we will figure out as we look at what everybody else does with the available information and the different types of models as they approach these large databases and the computational tools that they apply to them. But from the perspective of the National Toxicology Program, the priority setting as I see it is multi-phased priority setting. What I would like to see us do is, for each chemical that we do a broad spectrum of screening assays for, whatever they might be, we are able to calculate a probability that this is an immunotoxin, a probability that this is a carcinogen, a probability that this is. So I want an actual numerical number that I can work with that tells me something. Then on top of that, and incredibly important in this, is that I want to be able to group chemicals together that have like activity. Not like activity for carcinogenicity, but like activity for the mechanism that drives the carcinogenicity. Because to me, that is the biggest cost savings we're going to get in the short term from these assays and these types of approaches. because then I take that cluster of compounds and I treat them like we treat the dioxins. I'm going to find the one I think, based on probabilities and exposures, is probably the most toxic. I might find one or two others. And I'm going to, first, I'll take the whole group. And I'm going to run it through zebrafish. So I'm priority testing for zebrafish testing. That's my first priority. Because once I run it through zebrafish, I can look and see if the response in the zebrafish is the same. It's uniform. Did they all respond the same? The activity profile we get from the screening tells us they should, and I put it in a fish. Do they react the same? If they do, I'm very happy. I've got a group that seems to react together. Do they react like carcinogens, if I'm looking at a carcinogen, that also had this same activity profile, known carcinogens? If they do, even better. I'm happy. Now I've got a group of compounds. Then I'll take it one step further. I'll take the most potent one or two, and I'll test them in the more expensive definitive assays. I might define those assays very carefully based upon the mechanism I see, but I'm going to test them in the definitive assays. And once I prove in the definitive assay that, yep, it was supposed to be a carcinogen, it came out of carcinogen, we might not know the total mechanism, but these look like they all work the same, that's going to go two ways out my door. One way is I'm going to dump it on EPA or FDA, and I'm going to say, deal with it. These look like it's a group. They look like they all work the same. Tell us what else you need to regulate them, but as far as NTP is concerned, we're going to wait to hear back from you. The rest, it goes out my research door. Tell me why this cluster of activity caused this carcinogenicity. And we're going to get grants to look at the question. So when I think of setting priorities for this, that's where the NTP looks at the priorities. To me, that's a very logical, reasonable approach to using the information. And I think it's going to answer many of the questions that Gail laid forward in terms of how we want to move forward with this.
Additional comments on this particular aspect? Tina?
This is beginning to remind me a little bit about years ago when computer searches for literature first got started, and I used to always use Science Citation Index, and I would always find a seminal article and work from that, and suddenly you could put in a keyword and you could get tons and tons and tons of articles, some of which had absolutely no relevance. But one of the things that I'm a little nervous about here is we now have this exciting new tool, and that enables us to prioritize things by hazard. And I think we should remember that there's another aspect to this, and we probably want to also consider exposure in setting our priorities for testing. And, you know, you were saying you want to know the probability that something is immunotoxic. Is there a second half to that, immunotoxic at the levels that people are expected to be exposed to? Or is it, you know, just at any level? And for example, the EDSP program currently, that list of 63 chemicals, was totally prioritized based on exposure, not on expected hazards. So I think, you know, as we go forward, we sort of want to balance those two, and not just because we have this easy way to prioritize on hazard, just use that. We want to do a check step where we get into exposures too.
The NTP has always used exposure as part of our priority. I didn't bring that into this, but of course, we don't test things that nobody's exposed to. That said, I'm not going to tell you in advance that just because in the in vitro assays or whatever we're using as our screening assays, just because in those assays we only see response at doses that appear to be difficult to achieve in humans, we will not test those compounds. For all the same reasons we've always tested compounds at higher doses in animals. It's a question of ability to see the effect. And as I mentioned earlier, I still don't understand the dose response patterns I'm going to see in some of these screening assays and what they may mean in terms of overall response. That's part of that overall learning curve. As we get 2, 3, 4 of these down and we've worked through them, we can then go back and start thinking about, did we learn something about dose response here that's important that we can also play into this overall picture? I think it's a long-range learning curve.
So, Lauren, a brief comment?
Yeah, I I just want to clarify, for example, for the endocrine disrupting program, as you move through tier 1, that's going to give you some indication of hazard. And so instead of waiting to provide information to the public about what you think about degree of hazard, you might assess that at that point and also move them through your tier 2. That's all.
And that's a great segue into the last discussion point that we wanted to have this morning. It has to do with what needs to be communicated to the public about the issues that we've been talking about today and the state of the science, and at what level do we talk about these things, and how do we manage expectations around the science as it's developing. Trey, do you want to just make some opening comments on that, and then we can make that our last topic for discussion?
And again, many of the comments I had have been covered by Daniel and Gail in particular. Coming from the CPSC, for those who are not familiar, we are a small regulatory agency. We have less than 500 employees, but we have very broad regulatory authority over approximately 15,000 products, many of which contain chemicals, everything from household chemicals to children's toys to textiles, furniture, and can be a source of exposure. So this is very important, and I really appreciate being able to give CPSC's perspective, because oftentimes, again, being small and we're sometimes overlooked. And one of the things that we're considering are the new tools that can be used. So again, this was really a great opportunity to sit among the experts and hear the discussion about these new tools. Again, we're considering the use of in vitro methods and silico methods in the way that we approach hazard and risk assessment. Again, Daniel brought up nanomaterials, and I think that it's a good, example of a challenge for us. It's growing very rapidly. There are many products on the market that are reported to contain nanomaterials, and there's still a lot of questions about even the definition of nano. There are issues in terms of toxicity testing as far as size, the coatings, the agglomeration. If a product agglomerates, is it still a nanomaterial? And I think the federal government has come together and identified many of the data gaps that are there. And I think both the US and certainly in Europe, I think alternative methods have been pointed out as, is this an opportunity for us to really use alternative methods, in vitro methods, again, and silico methods to address many of these data gaps. Again, there's also pressure to reduce the number of animals. So I think that, again, this question of, for someone who's involved in toxin risk assessment, who's not an expert, how reliable are these tests? How should we use them to make decisions, and what do we say to the public? Now, some other considerations, kind of taking a step back, is the whole issue of public engagement, I think, has really changed in the past 10 years. With the advent of email, the internet, it's very easy to spread information and, unfortunately, disinformation. I've even had, just a quick aside, a private citizen talk about risk assessment on YouTube, and it was really bizarre to see someone discussing this, on YouTube. So we get a lot of public comments, and again, I think for us, it's where the rubber hits the road. Many people ask, is it safe? Is this product, what are the exposures, what are the risks? So it's really critical for us to get it right. Also, we're considering using Twitter and some of the news. So again, it's very easy to spread information. Also, FOIA. A lot of groups and individuals are FOIAing our what we're doing, our information. So a lot of what we do is out there in the public. So I think there's a great deal of scrutiny in the way that we approach, all the way from the hazard assessment all the way through the risk assessment. So I think we have to be very careful in the decisions that we make and the tools that we use. So I just point that out as when we're talking about, again, the decision making and what we base our decisions on. Are those tests, and specifically in silico methods, are they appropriate? Again, Gail has talked a bit about that. Again, are these tools valid? Have they been validated? Certainly, I think it's important what may be appropriate to talk to the public about, that we do have new tools, that there are, beyond the traditional animal testing, that again, we have in vitro and silico methods. But again, the only caution that I would have is if we try to draw conclusions, let's say based solely on computational methods. If we say that this is toxic or give some indication of its toxicity, is it appropriate, to use to disclose that information and to talk about that with the public based on in silico methods? And I think right now the consensus is, we're not there. But as we move forward, and someone has a study and says, this is a neurotoxin based on, is that appropriate? Again, is there some way, if it's in a peer-reviewed journal, yes, it's peer-reviewed, but is there some other layer of mechanism that can say that, yes, it is appropriate for us to use, particularly if we're not experts in this field, to use this information to convey to the public what the potential toxicities of a compound may be? I think those are important considerations when we talk about public engagement.
Thanks, Trey. Tina?
I have actually a question and a comment. A question to you, Trey, and I guess Gail and maybe Tina. Everyone sort of brought up that we don't know exposure, and you said that NTP uses exposure in your prioritization. My question is how? I mean, recent experience with CPSC, we found that our understanding of predictive exposure is really very, very low, very, very minimal. And Richard alluded to the really infant effort within that program to start that ex-post CAS as a commensurate activity. So when things happen, and even if we get an answer that's relevant from a hazard perspective, we really don't know in most cases down the chain of commerce what to say about exposures. And to just sort of dismiss it, I think, is going to be a mistake that is going to come back and haunt us. So that's, do you want to?
Yeah, because I think that's part of the equation for us is, again, and when I talk about products, someone's using the product. And again, when you're dealing with the public, it's different when you're dealing with scientists. So much of the question will be, will we use this? So as a scientist, you may say, well, the toxicity of this particular compound is not that great. But to the public, if there's exposure, if it's widely used in commerce, then, yeah, how does that enter into the equation? As an exposure scientist, that's one of the questions, too. So I think as we talk about prioritization, yes, what are the public concerns? What are the potential exposures? How does that fit into our decision-making process in addition to the toxicity and what assays are available?
And how do we even come up with a way to know what those potential exposures are down the chain of commerce, which is very, very different at the point of manufacturing? And the one sort of additional sort of question is, when we find information through testing and then the large epi studies find something completely different, and those information usually come out fast through the public domain because they get published in journals and because they involve human populations, they get picked up by media, then how do we reconcile the often disparate information that comes out?
So, Tina, I don't know whether Marilee put you up to this, but remember our next workshop for everybody is on the exposome. And that's coming up in February 25th and 26th. Again, I think these are important issues that we have to really grapple with in terms of understanding the exposures that might sort of direct some of our actions here. Lauren and then Gail.
Yeah, just a quick point regarding coming up and seeing different endpoints. I think that the toxicity, what we're talking about in terms of assays, enable us to really understand if those are really different outcomes. You see different endpoints following the same mechanism. And so just offering that, but we need to invest in that. Exactly.
One of our priorities at NIOSH is we also look at exposure and the number of workers that are exposed to a particular chemical. And, obviously we're, for funding, for internal funding, they want to see that you're looking at, lots of workers. NIOSH has an ongoing surveillance program where we try to get those numbers. It's not as robust as we'd all like it to be, but we do get some numbers as far as the number of workers. We also look at say in pesticides, number of pounds applied, and and we know which where they're being applied at, and can estimate the number of workers through that through that mechanism.
Thanks. So I have Tina, Carl, and Richard, and then we're going to give Yvonne and Chris the last word before we wrap this up. So Tina?
Just quickly on exposure and then also on communicating with the public. Exposure isn't, we always try to incorporate exposure, and there are issues with exposure estimation, just as there are issues with hazardous estimation. And we've got lots of different projects going on, and there's a lot of biomonitoring and reverse calculation going on, and it's its own area. And since I'm not a toxicologist, I tend to delegate it to people around me that know more about this area, but there's just as much going on in this area. I am a toxicologist, I'm not an exposure person, but in terms of communication, we've been communicating all along with the public about Tox 21. When the NAS report came out, we reported it we have twice a year, we have a federal advisory committee called the Pesticide Program Dialogue Committee, and we talked about it, and we set up a work group of the Pesticide Program Dialogue Committee. And so there is always, at our regular times when we get out there and talk with our stakeholders, and there can be public participation, we sort of try to keep people informed as you go along. And I think the idea is to keep people, we also have a website where people can go and sort of find out what's going on. So I think it's important to keep people in the loop, but not to promise things, or you have to. We're talking about managing expectations, and the only way you can do that is if at least you tell people where you are now and sort of let people know where you're trying to go. And so since we hadn't really talked very much about that, I at least wanted to talk about what the Office of Pesticide Programs had been doing.
The communication aspect, which I think echoes what Tina said. First, we've got an entire experience in literature on risk communication, and many of the lessons, I'm sure, are applicable here. But, explaining uncertainties, limitations, what we have, what we don't have. But one of the things other points I wanted to make that I think came out throughout the entire meeting, and that is this. There's a whole, and I hadn't heard the word mentioned during the discussion, at least not while I was in the room, but it's the concept of defaults. Our entire current approach to risk assessment has embedded in it a whole set of defaults which we have come to expect and understand and accept. And if you're going to move to an alternative method, whether it's CompTox or using microarrays, what have you, there's an entire new set of defaults that somehow it's not going to give you the answer, at least not in our lifetimes. And so built into that will be a set of uncertainties, a set of default assumptions, which have to then become sort of recognized and accepted by the scientific community, so they become sort of part of the new approach. And it's an interesting concept that I don't think we've given enough thought to.
Thanks, Carl. It's a good point. Richard.
One last comment about exposure. So prioritization actually has two pieces of it. So, as with the EDSP program, that was prioritized based on exposure. My little made-up scenario, we're going to prioritize based on hazard. But in both cases, the prioritization tools are imperfect, and so you almost have to have an iterative procedure where you do some simple but hopefully relatively high-quality approach on both, and the things which tend to bubble up to the top on both lists, then you have to refine. And you have to refine on really understanding where exposure is, refine really understanding where the potential for hazard is.
Thanks. So I think that from the discussion this morning, you can see that there are quite a number of issues that we can grapple with as a community and as federal agencies who are going to have to make decisions in this emerging science. There's also a natural tension between wanting to get the word out about advances in the science in order to continue to elicit funding for these kinds of activities, and at the same time managing expectations as to how those are going to be able to be used in decision making when they're in their early stages. And so I think these are some issues that clearly have benefited from the kind of discussion that we've had today. Yvonne, you are sort of responsible for this workshop, and I think we need to give you the the word here, and then Chris, as the sponsor here, perhaps you can make a final comment as well, and then we'll kick it back to Marilee.
Well, thank you, Bill. And again, I would like to share my time with George, who contributed greatly as well to, co-organizing this workshop with me and the rest of the committee. So I hope he will have a couple of words as well to say. But I wanted to first of all express how gratified I am with the outcome of this day and a half. And I think the credit goes to the liaisons and to the committee members who worked on this, to the speakers and the panelists. And I think we had a really invigorating discussion. I think we all had a very frank exchange of ideas, and I think we'll come out of this recognizing that computational toxicology is emerging science and it is highly relevant to decision-making process. So I hope that this was not only stimulating, but also will be followed up in some of the decisions that the agencies are making and some of the directions that people who are doing research are taking. So again, I would like to thank everyone for very lively participation. And again, perhaps George will have a couple of comments as well.
So I'd like to echo Yvonne's thanks to all the speakers and all of the participants. I think it was a really informative couple of days, and really liked getting the feedback from everybody. As I said yesterday, I am an optimist. I think that there's a lot of smoke that got that got generated in these discussions about and a lot of expectations that we're not ready to meet. But I also interpret that in a way that I think we're all desperate for new and better ways to assess toxicity in a way that's useful to public health. I also interpret this particular merger of computing power that we've never had before with life science technology that we've only had for a matter of 5-6 years is really poised to address some, not all of the questions that we brought up today, some, not all of the needs, but in ways that we've never had before. I think that we have great potential. I think our challenge now is to meet some of that potential in a way that shows people what the real worth of this, while managing the expectations in a way that is honest about when people can expect these technologies to come online in an applied way for regulation and public health.
Well, I want to thank the organizers for doing an excellent job. When NIEHS put together this standing committee, our goal was to open this type of dialogue, and I think today we have been extraordinarily successful in opening that dialogue between the government liaisons, the people who are making these public health decisions, and the scientists who are trying to bring a technology forward such that there's a better understanding of what is coming, when it's going to get there, and what it will mean. And I think that's an extremely valuable exercise. And so, again, I want to thank you for doing an excellent job. I want to thank all the members of the committee for their efforts, and I want to thank the Academy for putting it together. Thank you very much.
I want to thank you as chair of this committee for everybody, for their input, George, Yvonne, Chris, all the speakers, all the participants. It was a great 2 days. Let's continue. I want to offer a final word. I'll take this prerogative to offer a challenge to our federal agencies and the groups working in this interesting area of computational toxicology to continue this communication. I've heard the last several days that the most important thing that we can do right now is continue our communication, to collaborate, to form partnerships, to move this area forward. And we have to show some leadership in this area to do this. And I think it's a challenge to our federal agencies, whether it's through NIEHS, whether it's the EPA, to continue, in some cases, start working together better to move this area forward so that we can have a more efficient, cost-effective way of protecting public health. And without working together, we're not going to be able to do this. So it's a challenge to the agencies to continue in this process. Mary Lee.